Yeah. Hi, good morning, Kishorji.
How are you?
I am fine. How are you?
I am fine.
Hi, team, everyone. Good morning.
Good morning, sir.
Good morning, sir.
Good morning, sir.
Good morning, sir. Good morning.
Good morning, sir.
Good morning.
Good morning, sir. Subhash here.
Morning, Subhash.
Good morning.
So those who are
How many
comfortable.
people are there?
Yeah.
Morning, Ramlu.
Morning.
Good morning, doctor.
Good morning.
Good morning, Ramlu, sir.
Morning.
Hi, Prasanna. Good morning.
Good morning, sir.
Prasanna, I have sent the slide deck of today's
Okay.
over
Let me
mail.
just-
So since they were lying on the different laptops, so I have collated. So you will find that number 4 is a repeat. Actually, number 4 is 5, 6, 7.
Okay.
Okay, so 1 and 4 are in one slide deck. Then 1, 2, 3 separate. 4 is with 2, and then you read 5, 6, 7. Instead of 3, 4, 4, 5, 6.
Okay.
3, 5, 6, 7. Otherwise,
Right.
at the end, when we will complete this thing, now all modules I will correct the naming and then I will send it again.
Sure.
Because what happened now when we prepare for this thing now, so a lot of changes are there, new updates are there. So today morning 4 o'clock I was updating the slides and new information incorporating.
Okay. Not an issue, sir.
Okay. So can
Great.
we start or we wait for some more people to join?
We will wait for some more time. I think.
Okay. Not an issue.
Anyone else left? I think we are. Maria is there. Just a minute.
Yeah, we'll wait for 5 minutes more.
Okay. Not an issue. Everybody should write their name instead of the name of device, because what we have planned that we will be having pre-evaluation and the interactive session. So
Yes.
everybody has to write their answer on the chat box. So we will get the score once the evaluation is done from the chat box.
Right, sir.
Like Moto G9 Power, he has to rename himself.
Yeah, I think everybody is there with name. I think only one, Moto G. So that's one thing. Guys, we can switch on the videos like also Wiki initially. So those who are comfortable.
Everybody should be comfortable for next 2 days. Sagar, please, can you set your camera? You are inverted.
Yeah, I'm trying to do that.
You are upside down.
Yeah, I'm trying to make it vertical.
Sweetie, can you refocus your camera because you are not visible fully. It will be a
Guru
two-side. It will be a two-side session. It will not be that one-side session. I am reading and explaining the things. Everybody should have a lot of questions during the session so that the things are assimilated nicely.
Absolutely, sir. I think. Okay. One or two guys are left, I think, now here.
Okay, not an issue. We will
We're
start at
around
9:30.
35. Yeah, we will start at 9:30 sharp. I think.
Moto G9 Power, please rename yourself.
Sir, I'm trying actually. I'm searching. Can anyone help me, please?
You click on your, you have 3 dots there on your icon. Can you tell me, you can see the 3 dots.
Dharmesh, is this you?
What
Yes,
is his
yes,
name?
no.
Can you tell me your name? I can rename you.
Dharmesh.
Dharmesh. Okay.
Dr. Saxena. Piyush here.
Yes, Piyush, good morning.
Good morning. How are you?
I am fine.
So for 2 days, we all are in your hands.
Yes, definitely.
Hi, Piyush, morning.
Hi, Ramlu. Good morning.
Good morning, sir.
Good morning, everyone. Hi, Rakesh.
Good morning, sir.
Good morning,
Morning,
sir.
Rakesh.
I think everyone is here. Shall we start then?
Yeah, everybody has. Yes.
So now you have taken note, na? Everybody has joined, na?
Yeah.
Okay, fine.
Okay. So let us start the introduction of each and every one, so that I will be having an idea that the background of each and every person. So I am Dr. Nishod Saxena. I think everybody in your organization knows me very well. So this 2-day seminar will be conducted on majorly on the workflow of the biologics, vaccines, and injectables, including the single-use technology in the biologics domain. And with me, that my colleague, Ms. Khevna, will also be taking some of the modules. So I hand over Khevna to introduce herself.
Hi, myself is Khevna Majmundar, and I'm working as a director in Pharma Knowledge Center. And I have completed my MSc in microbiology and MBA. And I have 18 years of experience in pharma and biopharma companies. So this is all about me.
Welcome, Dr. Saxena and Ms. Khevna. I think thanks for your time for this training session today. So let me take this team here. Okay, so we have Mr. Rakesh Agarwal, we have Mr. Piyush Jain and Ramlu Kohli. So, and our entire sales and marketing, as well as the service team, which is based, regionally, pan India.
Okay, so can everyone individually can give a brief introduction quickly?
Sure.
Hi, good morning, everyone. This is Ramlu Kohli, and I'm based out of Hyderabad. So I take care of the pharma business for Antilia Scientific. Almost I have 15 plus years of experience in pharmaceutical business.
Okay.
I am Rakesh Agarwal. I am not from the education background. I am a mechanical engineer, but I am in this industry now for over 20 years, and I manage the APAC operations for Antilia.
Hi everybody, I am Shekhar Vasist. I am a zonal manager based out of Ahmedabad. I experience handling the team of pharmaceutical across the Western, Central and Northern region. Thank you.
Okay, thank you.
Yeah, hi, this is Samir Tawde. So I'm taking care of product management of Masterflex pumps, tubings, and fittings, and the other accessories. Currently, with this organization, I'm associated with more than 15 years.
Oh, where are you?
In Mumbai.
Mumbai, okay. Can everybody can switch on their cameras for a quick time, short period of time, so that we can see?
So unfortunately, sir, my laptop does not have that camera. I'm looking for my external source.
Okay, okay.
Just wait
Not
for a
an
few
issue.
minutes.
Okay, okay, okay. So next one, please quickly let us, because we have short time.
Hello. I am Guru Prasad, based out of Vizag. Basically, I have done a master's in microbiology, and I have 15 plus years of expertise in biopharm.
Okay.
Hi, this is Maya, based in Hyderabad. It's almost 2.5 years now been associated with Antilia in bioprocess. The total years of almost 13 years experience in the market. And especially in bioprocess related, it's almost 7 years.
Okay. Arya, your microphone is disturbing. Can you fix it? Because it will be required during our entire session.
Currently, I'm just on my way to home. About another 15 minutes, I reach home.
Okay, okay. Not an issue. Not. So next.
Hi, good morning all. My name is Sunil Dasari. I'm based out of Hyderabad. I'm responsible for on-cam for pharma through Antilia Scientific.
Okay.
Good morning,
Hi,
sir.
I'm Sampath, handling key accounts. I'm associated with Colpharma since past 7 years.
Okay.
Yeah. At Hyderabad.
Okay. Okay.
Thank you.
Hello, good morning. Myself Jagruti Pirankar. I am product application specialist working in Antilia from last 2.5 years. I'm based in Mumbai.
Okay. Yes, Rajesh.
Yeah, I'm Rajesh. I am based out of Bangalore, and I'm from the service team. And I've been with Masterflex for 15 years.
Okay.
So we take care about installation, IQQ, related. Yeah.
Okay.
Thank you.
Good morning, sir.
Hello.
Myself, Ankit Bhavsar. I am based at Ahmedabad, and I am taking care of key account manager role based at Ahmedabad.
Good morning, everyone. Myself, Rakesh Tiwari. I am based at Vadodara and looking after key accounts like Sun Pharma as well as Alembic. So I have overall more than 12 plus years of experience, and in Antilia Scientific, I am working for almost 4 years.
Thank you.
Morning, sir. Myself, Vijay. I'm key account manager for Biopharma. I'm based out of Hyderabad.
Good, sir. I am Raj Shekhar. I am based at Hyderabad. I have been with the organization since 4 years.
Okay.
Good morning, sir. This is Shashank Samant here. I joined Antilia 2 months back as a zonal manager. I am based in Mumbai. And prior to this, I worked for Toshvin Analytical almost 15 years, handling Shimadzu instruments, analytical instruments. So overall experience is 18 plus years of experience in this industry. Thank you.
Okay, thank you.
Yeah, myself, Subhash here. So I handle the key accounts of Karnataka region, especially Biocon and Syngene. I have 14 years of experience in this field.
Okay.
Morning, sir. I am Dharmesh Parmar. I am handling Hyderabad region key accounts, Dr. Reddy's, Emil and Mylan. So I'm based at Hyderabad, and from in this industry, I'm from last 7 years.
Okay, okay.
Good morning, sir. I am Bharat Barate. I am from Pune and taking care of non-cam and BNC account.
Okay.
Good morning, doctor. This is Kaushik. I'm based out of Bangalore, and I take care of BNC accounts, BNC pharma accounts, and everything here in Karnataka, Tamil Nadu, and Pondicherry.
Okay.
Thank you.
Good morning, all. This is Sagar. I'm based at Mumbai, and I'm associated with Antilia Scientific from last 2.5 years. I take care of major farmers and OEMs in Mumbai and Goa.
Okay.
Thank you.
Good morning. I am Jigar Shah. I am based at Ahmedabad, and I am handling non-chem account for Gujarat for Antilia Scientific.
Okay.
Hi, sir. This is Pradeep Kumar from Chandigarh, based at Chandigarh. I have associated with the Antilia Scientific from last 3 months and take care of the non-chem accounts from North region, sir.
Okay.
Yeah, thank you.
Good morning, everybody. This is Ankit Saxena here on this side from Noida, Delhi, NCR. I have joined Antilia in the month of January this year only. Prior to this, I was working with Anton Par, and I have total experience in pharma industry for about 13 years. And academically, I've done BTech in biotechnology and also a management program.
Okay.
Hello, good morning. Atul here. Hello.
Yes, Atul, continue.
Yeah, good morning, Atul here. I am working from 1.5 years with Antilia Scientific in taking care of Madhya Pradesh region. Overall, I am working with the pharmaceutical industries and having experience around 10 years.
Okay.
Good morning, everybody. This is Nilay Mishra, based out at Guwahati, and I'm associated with Cole Pharma since past 2 months and having a total experience of overall around 10 to 11 years.
Okay.
Hi, good morning, everyone. My name is Pramod Survashi. I'm taking care of product management for Masterflex for industrial and research segment. Almost 17 years of experience in marketing. By graduation, I'm chemical engineer and MBA in marketing.
Okay.
Hi, good morning, doctor. This is Hitesh Tandale. I am based out of Mumbai, and I am associated with Antilia since 8 months, and I have overall 6.5 years of experience, and my education background is master's, and I did my MBA as well. So that's it.
Okay.
Hello.
Yes, yes.
Hello. Yeah, good morning, sir. So this is Devmalya Basu. Actually, I am based out of Pune, and I'm handling the key account like Serum and Inzin. And my experience in this organization is almost 6 years, and more than 6 years, and total experience is 14 years, close to 14 years. And my background is I did my MSc in industrial biology.
Okay.
Yeah, thank you.
Good morning, sir. This is Gopal Kulkarni. I stay based in Pune, and I'm working for the Cole Pharma from last 6 months, and I have total experience of 13 years in this industry.
Okay.
Good morning, sir. I'm Madhu Mohan, based at Bangalore. I'm handling the Syngene and Biocon as a CAM executive.
Okay.
Hello, good morning all. This is Manoj Salve. I'm working as a senior product specialist in English technology. I have done my B Pharmacy and MTech in Bioprocess Technology from ICT Mumbai. I'm based out of Hyderabad.
Okay.
Hello. Good morning all.
Working at
Yes, Tirupati.
Yeah, good morning. Yeah, my name is Tirupati here. I'm based out of Hyderabad. I'm having experience of 8+ years in the industry, and I'm working with Antilia Scientific from almost 1 year.
Okay.
Hello, hi everyone. This is Piyush here, and I work with my team on the India operation side. I am having a whole experience of 20+ years in the life science and diagnostic industry, and with the Antilia, I have been working with since last 15 years now.
Okay. Okay. Thank you, Piyush. So I think everybody is completed, so we can move ahead, or some people are left.
Prasanna, I could not hear Deepa here. If I have missed out.
Yeah, no, she's there. Deepa, could you?
Yeah, I'm just having a little trouble with my camera. One second.
That's fine. Yeah.
Hello, Dr. Nishod and everyone. I'm Deepa here. I'm a part of the Masterflex fluidic stream handling Saint-Gobain products and valueplastic connectors and other products. I have an experience with the company for the past 1.5 years now.
Okay.
I've done my biotechnology and also my MBA.
So I think anybody else is remaining for introduction, or we can move ahead.
Anyone left, guys?
I think we can move, Dr. Nishod. We can move.
Yeah. Yes,
Okay.
we can move ahead.
Okay. So as per the introduction, I found that many of the people have the background of the biotechnology and all these things. So this workshop is designed basically for the bioprocess in biosimilars, vaccines, and injectables. So some of the parts, some people may find that it is known to them, but they can take it as a refresher for this thing, because all people have to be given the insight of the process flows in the biological manufacturing and vaccine manufacturing. So before starting, we have a quick pre-training evaluation where I will be projecting my screen one by one. It is a multiple choice of questions. What you have to do, you have to write question number and its option, which you find the correct answer on the chat box, so that we can collect your answers and prepare a sheet for the pre-evaluation session. So now I am sharing my screen. Everybody is able to see my screen? You can give the
Yes,
reactions from the
Yes,
reaction.
sir. Yeah.
Okay.
Yes.
Okay. So you have to, everybody has to respond on the chat box for the question number followed by the option which you feel the correct answer. The question number 1 is, how many quality tests are performed in large molecules? Option A is 25, option B is 50, option C is 150, and option D is 250. So for you can start giving your answers in the chat box. Yes, quickly, because we wanted this to be completed. Please write your question number also, because people are writing only the option. So please write question number dash the correct answer. Yes, this is good now. Quickly. Okay. So shall we move to next question? Okay. Question number 2 is, molecular cloning can be done with the help of RNA, protein, DNA, and the D is all of them. Please respond on the chat box. Okay. So now let us, still people are answering. Okay. Still people are answering. Yes. Okay. Now let us move to question number 3. Number 3 central dogma is DNA, RNA, protein, RNA, DNA, protein, RNA, cDNA, protein, and the last option is cDNA, DNA, and protein. Okay, good. Now the answers are coming quickly. Okay. Okay. Now let us move to the question number. Still people are giving the answers. Okay. Let's move to next question. Question number 4 is harvesting of cell is a part of upstream, B downstream, C lyophilization, D cloning. Okay. 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 And now let us move to the next question. Question 5 is gel filtration chromatography works on A, size and shape, B, charge, C, hydrophobicity, and D, affinity. Okay. 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 Very good. It is good interaction. People are responding quickly. Okay. Now let us move to the next question. Question number 6. Aim of aseptic processing is safe, effective products that are free of microbial and other contaminants. B, increase the production. C, to make sterilized product, and D, none of the above.
Rajesh, I think, can you mute yourself? Thanks.
Okay. Okay. So let us move to next question. Question number 7. Following is not the method of sterilization. A, autoclaving, B, radiation, C, lyophilization, and D, all of above. Okay. Question number 8 is proteins are amino A 17, B 20, C 38, and D 149. Okay. Okay. Now next question. Question number 9. What is the difference between DNA and RNA? A is replaced by G, C is replaced by T, T is replaced by U, and C is replaced by U. Okay. Okay. Now next question is, proteins are synthesized at cell membrane, endoplasmic reticulum, ribosomes, DNA and RNA of nucleus. Okay. The next question, 11. Viral filtration is required in which of the following? Product manufactured using mammalian cell host, product manufactured using bacterial cell host, product manufactured using yeast cell host, all of the above. Okay. Question number 12 is, which one of the following is not a QTPP? None of the below: route of administration, dosage form, delivery systems. Okay. So let us go to the next question. What is 510K in US FDA?
Kedar, please mute yourself. Kedar.
What is 510K in US FDA? Okay. And the last question is, product yield is highest in batch fermentation, continuous fermentation, cell culture fermentation, and fed batch fermentation. Okay. Okay, thanks for participating. We will compile these data and then we will share your correct answers with you. So now let us move to our first module. That is course objective and comparison between small molecules and large molecules. Can everybody is able to see my screen or no?
Yes, we can see. No,
now you can see my screen.
Now we can see. Yes.
Okay. Yes. So from here we will start our webinar. So this webinar's topic which we have selected is technical training on pharma and biopharma processes, workflows for vaccines, injectable formulation, and single-use technology implementation and expanding the scope. So before starting the webinar, let us fix some of the housekeeping ground rules. And these are the, please put on your mobile phones on the vibrate mode. And if you have important calls, you can take during the break, because we have planned some of the breaks also. And make yourself very comfortable. Participate and have fun, because many things will be very basic things, but they are very important in biologics industry and vaccine industry. And questions are always welcome. You can stop me and put your questions, so that everybody who are present, they are benefited from the discussions. And keep ready pen and paper if you have any question to be end of the session or anything. And once we will complete our this workshop, we will be issuing a e-certificate to all the participants. So my colleague Khevna will be sending an Excel sheet where you have to submit your details, so that we can prepare the e-certificate and send it to you. So now the information within this presentation is based on the presenters and the views of the presenters for the purpose of a training workshop. The background is what? This program is targeted towards imparting theoretical as well as practical knowledge of process workflow of biologics, vaccines, injectable formulations, single-use technology implementation, and expanding the scope. After completion of the course, participant is expected to have in-depth knowledge and better understanding of industry guidelines, regulations, regulatory bodies, and compliances for the future prospects. The modules have been compiled to introduce the attendees to various aspects of basics of process workflow in biologics, vaccines, injectable formulations, and single-use technology. It benefits in assuring quality production. Several specialized modules have been added to guide the candidate through FDA and other regulations, compliance needs, comparative study, rules in India and other countries, etc. The curriculum is an admixture of components like theory of the subject, hands-on practical training, group discussions, case studies of EMEA and US FDA, and Q&A sessions. What are the objectives of this course? The delegates will learn and understand the process workflow of biologics, vaccines, injectables, and single-use technology. We'll learn and understand that how to apply current regulatory guidance to process workflow of biologics, vaccines, and injectables, and new development in the process workflow of biologics, vaccines, injectables, and single-use technology. Understand the requirement of the single-use technology and what are the challenges faced by this industry. Describe and discuss industry best practices and requirements for implementing single-use technology. Aseptic practices, personal health practices, gowning and trafficking patterns in the clean room with single-use technology. Cleaning, disinfection, and contamination control practices when we are working with the single-use or the biologics facility. Understand how to establish the best microbial control strategy. In addition, the course also covers the main elements of the contamination and its control system, and its documentation, training, and system monitoring and review. Now let us come to the first thing. What is the comparison between a small molecule and the large molecule? So let me give a very simple definition for a small molecule and large molecule. These small molecules, which are chemicals, the drugs or the medicines which are manufactured using chemicals are called small molecule. Very simple definition that the drugs or the medicines which are manufactured using chemicals are called small molecules. And large molecules are the drugs or the medicines manufactured using microorganisms. Yeast, animal cell lines, or the viruses. Why viruses? Because all vaccines are falling under the category of large molecule, which we will see at a later time. So vaccines are also large molecules. So large molecules are manufactured using microorganisms, and small molecules are manufactured using chemicals. This is the basic fundamental difference between these two industries, which are called pharmaceutical industry and biopharmaceutical industry. Now, what are the other differences? If we see biosimilars, we call it biosimilars. We will see in detail why we call it biosimilars. And in pharma industry, where small molecules are manufactured, we call it generics. And sometimes people call it biogenerics also. When it is biopharmaceutical, it is called biogenerics. When it is small molecule, it is called generics. So biosimilars or the biogenerics are proteins. They are unstable and they are non-immunogenic. Very important part, because this is one of the aspects biopharmaceutical industry has to do a lot of studies that my biosimilar is non-immunogenic. Whereas in small molecules, they are chemicals, small size, they are highly stable, and they are immunogenic. How it is immunogenic? there are many drugs which are used for immunocompromised patients. For example, oncology drugs, they are immunogenic, and the person who is taking the oncological drugs, they are immunocompromised. Their immunity is lower. We all have seen during this COVID that the advice from the government or the vaccines should be given first to the patient or the people, those who are having low immunogenicity, maybe because of natural reason or on some drugs. And which are these drugs? They are the generic drugs from the small molecules. This is the first difference between biosimilars or biogenerics or biologics and generics. Now let us see what are the other. Now the size of the molecules, the chemical or the generics or the small molecule medicines, they are very small in size. And if we see the biopharmaceuticals or biosimilars or biogenerics, their size is very big. We'll see how big it is. Let us compare with the molecular weight. Small molecules, their weights are ranging from less than 500 to 900 Daltons. What is Dalton? Dalton is a weight of one hydrogen molecule. And what is the weight of one hydrogen? 1. Because it has one proton, and that weight is called 1 Dalton. So the molecular weight of generic drugs is ranging from less than 500 to 900 Daltons. Whereas in case of large molecules, if we see, the weight of the molecule is ranging from 4,000 to more than 140,000 Daltons. This is the difference between the molecules. Now, if we go to the structure of these molecules, small molecules, their structures are very simple and well defined using many analytical techniques, NMR, mass, X-ray diffraction. The structure of small molecules is well defined. We know what are the components are there. But if we go to the large molecules, their structures are very, very complicated, complex, and they are having the structural variations also. For example, if we buy a paracetamol from the market, the purity of the product in terms of the structural differences, each and every molecule of the paracetamol will be same in structure. But if we buy filgrastim, erythropoietin, or pegfilgrastim, we are not sure that each and every molecule in that injection is same. Structurally, they are different, and that is very difficult to identify and what impact it is having when it goes for the marketing authorization to US and Europe. So this is the major difference. That is why this science is very complex science, which we are going to run throughout next 2 days. This structure of large molecules are very complex, and there are variations also. What is next? The manufacturing. The small molecules, the manufacturing processes are well defined, and we can make identical copy of the molecule using the same chemical process. Identical copy. But in large molecule, when we go for the manufacturing process, we are not able to identical copy, rather we are able to make a similar copy. The identical copy means structurally exactly the same, but in similar copy, structurally we are not sure that they are structurally the same molecules. This is the basic fundamental difference. That is why the term we listen or hear in the biomanufacturing industry, the biosimilarity. With what? Biosimilarity with what? Biosimilarity with respect to the innovator's molecule. And what is innovator? The company or the organization who has first time developed, tested, and proved clinically that this biological molecule is safe, efficacious, stable for a defined indication. So this is where the entire biosimilar industry is working. The next is approval requirement. Marketing, suppose we want to go for the marketing authorization. In small molecules, if they are generic, I have included all these different terminology used in the regulatory. If they are generic, because we are now talking about generic versus biogenerics, and they are proven molecules, what we need to do for application, we have to do the only bioequivalence study with the innovator model. That means a very small clinical trial with the bioequivalence is always done with the healthy volunteers. We need not to do clinical trial on the patients when we are talking about the marketing authorization requirement of small molecules. Only small bioequivalence study, that is called PK/PD study. Most of the cases, it is only PK studies conducted on healthy volunteers. But when we come to the large molecules like biosimilars, vaccines, we have seen all vaccines, they have passed through all animal trials, all phases of clinical trials. We have to do large clinical of actual patients. That is called phase 3 clinical trials. So this is what the difference in small and large molecules with respect to the approval requirements. There are a lot of other requirements for large molecules, which we will see later on. But in terms of clinical studies, this is the difference between the small molecule and the large molecules. Now, manufacturing quality test. In case of small molecules, almost 50 or less than 50 analytical tests, whether they are a simple test or the specialized test, are more than enough for quality check. But when we go for the large molecule, there are more than or equal to 250 analytical tests and complementary tests. One test should support to other tests, has to be performed on the molecules. That is why if you see the release time, generally in industry, the batch release is always on pressure. If you see the release time of a chemical molecule is hardly 5 to 7 days. Now I will differentiate biologics in two categories. One category is biosimilars. The other category is vaccines. If you go to the biosimilars, you have to give assay value. In case of small molecules, 99.99% assay is done on HPLC. But if you go to the large molecules, the assay is done either in vitro or in vivo. In vitro means the assay is performed on the cell lines in the laboratory. And in vivo means the assay is performed on the live animal. For example, erythropoietin. Erythropoietin bioassay is done on mice. So the time period to complete this analytical testing is very high in terms of biologics. Now, this is the case of biosimilar where you need small molecules, biological assays are hardly done for any molecule. But in large molecule, each and every molecule is having a bioassay where either in vitro or in vivo method is used. Now, if I come to vaccines, the releasing procedure, there is a mandatory requirement in India. That is why what happened in the month of April, May, when there was a second wave and the non-availability of the vaccines. The main reason for the non-availability of the vaccine in the month of March, April, May was that each of the vaccine manufactured by each and every manufacturer needs to be tested by a Government of India laboratory located in Kasauli. Each and every batch is sent to Kasauli lab. They are having their animal models and the other chemical testing facilities. And each batch is tested for that bioassay based on the assay described in the marketing authorization. And once the Kasauli central lab is releasing that batch, the manufacturer is able to release the batch in the market. So this is what the complication in the testing and releasing of biologics and vaccines. The next is how these drugs, small molecules and large molecules, are administered. Biologics drugs are majorly injected or infused, directly going to the bloodstream. And small molecules, they are mostly taken by oral route. Because why this is the reason, major reason? Because these small molecules, they are sustained by the digestive system. The enzymes present in the digestive system, they are not modifying or degrading the small chemical molecules which we are taking through oral route, whether it is a tablet, capsule, or oral liquid. But if we take any of the large molecule which are mainly made up of what? Proteins. They are getting degraded or denatured in our digestive system by the enzymes, digestive enzymes present in the digestive system. That is why the mode of administration of all large molecule is either intravenous or intracellular or intrathecal or by infusion. Though the small molecules, majority of the molecules are taken orally. So these are the differences in the small molecule and large molecule. Now, anybody is having any question on this part? Please let me know.
Doctor, is there any, means in large molecules, there are any types, or is there something like that?
Types means?
There are any further category in that?
Large molecules.
We call biosimilar, so because there are various microorganisms.
There are two categories. There are two categories. One is we have divided into vaccines and biosimilars. Now, biosimilars, they are having different generations. For example, I can give you filgrastim. Filgrastim is a first generation biosimilar. And all biosimilars, these proteins are found in human body. They are not the synthesized or these all molecules, those who are falling under the category of biosimilars, these proteins are available in human body. They are synthesized in the human body. For example, filgrastim, erythropoietin, interferon alpha 2b, and these monoclonals, rituximab, Ambril, they all are available in human body. But when we develop the molecule to higher stability or higher performance, the second generation molecules are coming. For example, filgrastim, if we take, whenever there is a chemotherapy for any patient, he has to take 6 injections of filgrastim to boost his immune system. But what has been done, that the filgrastim, which is a natural molecule, is re-engineered with pegfilgrastim, polyethylene glycol. It has been added to the this filgrastim, and what was the advantage? The sustainability in the body or the clearance from the body of filgrastim has been increased. That means the half-life of the same molecule in the body has increased by 6 times. So instead of taking 6 injections of filgrastim, only 1 injection of pegfilgrastim is required. So pegylated versions of biosimilars, they are called second generation biosimilars. They increase the half-life of the molecule in the body. Otherwise, there is no further categorization or classification of biosimilars. They are biosimilars. Someone else has innovated that product, and rest all manufacturing companies are making similar products which are called biosimilars. So we have biosimilar for filgrastim also and biosimilar for pegfilgrastim also. The Roche has developed filgrastim and Roche has developed Neulasta, which is called pegfilgrastim. So Roche is innovator for filgrastim and pegfilgrastim, and Intas, Zydus, and all other companies, those who are making similar copies of filgrastim and pegfilgrastim, they all are biosimilar. And two companies biosimilars are not similar.
Dr. Piyush here. So I think another thing I think what Ramu is trying to understand probably is the biosimilar workflows. So when we talk about the biosimilar workflows, whether the MABs or the mRNA or CGT, all these are different workflows of under the biosimilar, or they have some different category altogether. Am I right, Ramu?
For all these, these all are different platforms. Like, let me explain the platforms.
Sure.
When we ultimately all are proteins. Ultimately all are proteins, but the manufacturing of that protein can be done by many platforms. For example, we can use the bacterial platform. We can use the yeast platform. We can use the cell culture platform. In cell culture platform also, we have different cell lines like CHO cell lines or the Vero cell lines. So we will see actually the workflow of manufacturing of biosimilar, which is ultimately a protein, is same. What you have to do, we will see in next slides what is the workflow of any protein to be manufactured using different platforms, whether it is a bacterial platform or the mold platform or the cell culture platform or the new technologies which has been developed. For example, Moderna is a mRNA vaccine, or our this Pune-based company, they have developed mRNA-based vaccine. Zydus Cadila has developed what? The DNA vaccine. And if you see Serum Institute's vaccine or the Bharat Biotech's vaccine, they are cell-based vaccine. So what they have done? The same process. Bharat Biotech, Serum Institute, Bharat Biotech using Vero cell line to manufacture that protein, which can neutralize that COVID-19 virus. Serum Institute using CHO cell line to manufacture that protein. Moderna messenger RNA vaccine, what they are doing, that messenger RNA, which is responsible for manufacturing that protein, is injected in our body to manufacture that protein. Instead of in vitro manufacturing the protein, Moderna or messenger RNA based vaccines, they are giving messenger RNA to human being to manufacture that protein. And what is DNA vaccine? DNA is one step behind. That DNA is injected in our body and DNA is making messenger RNA, and then messenger RNA is manufacturing the protein. So it depends upon different platforms from where we are using to manufacture the ultimate protein. These are the differences.
Thanks, sir, Doctor. I think,
yeah. Dr. Nishad Subhash here
yes.
From Bangalore. I just like to understand what are the different kinds of large molecules, is it a vaccine or insulin? So what are different kinds of large molecules, and can we also go through what is the process of manufacturing for this different types of molecules?
You see, we have this divided into vaccine and biosimilars. So in biosimilars, all platforms are available, that is E. coli platform, yeast platform, and the cell line platform. Now, vaccines, we know, they are whole life cell vaccine, attenuated vaccines, DNA vaccines, messenger RNA vaccines. If we come to the biosimilars, if the protein is manufactured using bacterial cell line, it will not be having the glycosylation, because that enzyme is not available in E. coli plate. So any molecule, protein molecule like monoclonal antibodies or darbepoetin or erythropoietin, if you see the structure, apart from amino acids, apart from the protein chain, these protein chains are also having the glycosylation, that carbohydrates. Number of carbohydrates are attached to the protein. So if we are using E. coli platform, it is due to the lack of that enzyme, the glycosylation is not possible. So we are not able to manufacture the monoclonal antibodies in E. coli or yeast cell lines. When we come to the glycosylated protein, the monoclonal antibodies, they are manufactured using CHO cell lines. So these are the differences in different biosimilars, the structural differences. Any other question?
Yes, doctor. This
So
is
Dr.
Gopal here.
Samir here. Yeah,
Yes,
Gopal,
sir.
go ahead. Then
Okay, thank you. So I would like to ask that what are the differences in new drug molecule compared to generic medicines? So what are the different tests are there which segregates the generics and new drug molecules?
Gopal, I have made another slide deck for all these NDA, ANDA, IND, and PLA and 510K. So can we discuss that slide deck?
Okay, sure.
Okay, because this is a long discussion.
Yes.
Hello everyone. So right now, whatever we are discussing, whatever Dr. Saxena is sharing on the content side, I would appreciate if we keep our questions related to it only, because a lot of content is still to come, right? So let's not jump the gun.
Not an issue, Piyush, because these questions will come. So I know what is the content coming in next slide deck. So the same way I will pause so that we have a track. What are the questions coming to the mind? I have one slide deck where I have explained IND, NDA, ANDA, BLA, and 510K. So there we will discuss in detail what are the differences in these 3, 4, 5 kind of marketing authorizations. Yes, Samir, you was asking some question.
yes, doctor. So the biosimilar medicines or drugs, those are equivalent to biological products, or it's a different?
You see, these are the two different terminologies. Biological products, I have given a very clear definition. Any biological product is manufactured using a bacteria, it can be a yeast, it can be a cell line, it can be a virus, or it can be a messenger RNA or DNA, till the messenger RNA, DNA are live. So biologics is a biological product. Now terminology used biosimilar. So why we call biosimilar? Any biologic product, for example, corona vaccine. If we take corona vaccine, this is also a biologic product. But no as a biosimilar, because this is the every company, whether Pfizer, Moderna, or Zydus, or Serum, or Oxford University, or Bharat Biotech, they all have innovated a new product. So all are falling under the category of biologics or biological product. When a company wants to develop a product which is already existing in the market by another company, and most important part, it has completed its patent life cycle of 20 years. For example, rituximab, it was developed 20 years back by one of the company. They have identified the protein. They have developed the proof of concept. Yes, rituximab is a good, can be a good candidate for rheumatoid arthritis. Then they have done some of these studies to identify that protein, isolate from the blood of the human being. Then they have done the protein sequencing, structural identification. They have done the related gene sequencing, which are the gene available or responsible for manufacturing this protein. They have constructed that gene, inserted in a vector, inserted in a host, optimized the growth expression, then scaled up, then did a preclinical phase 1, phase 2, phase 3, and then they have obtained a marketing authorization in 10 years of time. That is called innovator product. And what is biosimilar? I am having rituximab available in the market. Now I have to reverse engineer that protein available in the vial. And I know from the literature that what is the gene. I can buy that gene from the market. I can buy vector from the market, insert that gene into the vector. I know the CHO cell is very good cell for the growth of this monoclonal antibodies, because it is having a lot of responsible for glycosylation. So I have all the knowledge and the final product available in the market and trying to make a copy of that original product. That is why we call biosimilar. Biosimilar does not mean exactly same. But I don't know, my gene sequence will be same because it is available from the innovators literature. But I don't know which vector he has used, which host he has used, what are the media component he is using, what kind of reactors he is using, what kind of downstream he is using, what kind of chromatographic techniques he is using, and what kind of analytical procedures he is using. That is why my product is similar but not exact. That is why we call it biosimilar to the innovator's product. And innovator in last 20 years, they have developed the manufacturing process, optimized manufacturing process, scaled up manufacturing process. They have started using glass reactors, gone to the SS reactors. Now they are using 10,000 liter single-use bags. But today when I am developing, I am using my SS reactors only. So that is the difference between biologics. Biologics means any medicine originating from microorganisms. But biosimilar means already existing product, biological product from an innovator company which has completed its patent life cycle. Before patent, I cannot manufacture and sell that product. Yes, I can start the developmental work. Yes, Subhash, you have any question?
Yeah, so apart from biosimilar, we also have bioidentical manufacturers in India.
Bio?
Identical means they follow the same process also. Is there anything like that?
Bio, I am not getting your bio.
Bioidentical means even the process, will they follow the same?
It is not possible throughout the world.
One more question, like these biosimilars are applicable up to the patent age or only even after the patent age?
Biosimilars are only allowed to be marketed after the expiry of patent. Only the expiry of the patent, that is 20 years. And bioidentical is also, you see, I have one slide where I will show if I call biosimilar, bioequal, bioidentical, the twins are similar or identical, but they have their genetic makeup are same, but there are some certain differences. So that is called biosimilar or bioidentical or follow on biologics भी बोलते हैं उसको, follow on biologics. Shekhar, you have any question?
Yes, doctor. As you rightly said that somebody can make biosimilar after the, you can say, period of 20 years. So by then the next competitor can start their analytical works after 5 means remaining 5 years or something, or it's some timelines there.
No, you can to wait for the expiry of patent to start your work. And waiting for the date of expiry.
Okay.
The next day, if your products are ready, you have done all these studies, developed the data.
Yes.
You can launch your product next date of expiry.
Okay. Fine. So means they can do the all groundwork or
everything you that is what is done in this biosimilar. No? Everybody has started long time back. You see Vocard and this Zydus. Zydus is almost 30 years old, 30-40 years old biologics division.
Yes.
They have started their R&D, cloning, isolation and everything long time back.
Means they have targeted some molecules already and they have started working 20 years before already.
Yes.
Fine, thanks.
Okay. Dev Malia, you have question?
Sir, doctor, I do have one question. That is, if I understand correctly, so for biologics or biosimilars, the process may vary. If I say if lupin is producing one hormone and serum is producing one hormone, the manufacturing process may be vary, but the product will same, and the quality of the product will also may vary. So can I say that?
No. Please let me clear. In innovators product and in any of the biosimilar, only one thing will be common or same, exactly same, is the gene of interest. Because we know that the sequence of that gene is known, or the amino acid sequence of that protein is known. These two are the basic fundamental for developing any biological biosimilar from the literature of innovator. Now, based on either from your amino acid sequence or from the genome sequence, you design your genome and then make a host, but rest everything is different. You don't know what is happening there and how this has been evolved at innovator's place. So the ultimate product will not be exactly identical copy or the same. It will be a similar or a twins. There are some differences which are not physiological effective, clinically effective. but there will be some differences in your molecule and the innovator's molecule.
Okay, okay, sir. So the process for different company, the process needs to be validated or needs to be patented also for biosimilars. So suppose Biocon is producing insulin and Lupin is also producing insulin in a different way. So that process needs to be patented or it's not required?
Yes, there are two terminologies you have used. One is validated, other is patented. So every company, they are investing, except from the innovator. Innovator has spent a huge time and money, but biosimilar companies, they are not using that much of money and that much of time. So every company, if they are developing a new process, every company is creating its intellectual property. That means they are getting the patent. So every company's process is patented because this is the intellectual property of the organization or to protect their processes. Second terminology you have used, validated, means the process which this session we will be having tomorrow, process validation, but for a brief understanding, any process developed by any company or used by any company, until unless it is valid, we have a case study also on this. Until unless it is validated, validated means the consistency to show that the same process will result in the same product every time. So every company has a regulatory requirement that process should be validated, and process validation is a GMP activity. It has nothing to do with the patenting of the process. Sagar, you have any question?
Thank you, sir.
Yeah, only like, is biosimilar and the biogenerics are the different things, or?
You see, what is generic drug? Generic word has come from pharma industry, where we manufacture the small molecules. So what happened in India or other parts of the countries? New molecule, that means that molecule is not existing in the world. Developing a new molecule for any of the disease is a very long time and very expensive thing, and success rate is very low. So to grow the pharmaceutical industry, the manufacturing companies or R&D companies or the contract manufacturing organization or development organization, what they have done, all these small molecules, those who have gone for off patent, unka patent khatam ho gaya, they have developed some process of manufacturing the same molecule in pharma industry. And now you see paracetamol is manufactured by at least 1,000 companies. Ibuprofen is manufactured by almost 150 companies. Everybody has having the different source of raw material, different equipment, different processes. Ultimately, they are manufacturing 500 mg paracetamol tablet. This kind of development of drug is called generic drug. And in generic drug, you cannot sell with a brand name. That is what Indian government is behind. You have to sell with the generic drug. You can say paracetamol 500 mg tablet. This is called generic. That means somebody else has innovated that drug, sold in the market for 20 years. Once the patent has expired, other manufacturing companies has started manufacturing using raw materials from different sources, different equipment, different processes. Ultimately, the product is having same bioequivalence. Same bioequivalence. This is called generic drugs. And that has grown the pharmaceutical industry worldwide, including India. India is number 3 in generic drugs. Okay. In terms of volume. And number 10 in terms of value. Because we have a large manufacturing capabilities. So we have a highest volume. But our cost of production is very low. That is why our strength is 10th in terms of value. Now, same terminology has been used by some people for biologics product, since they are the generics of the already existing expired patent products, so they are calling it biosimilar. This is what the biosimilar. Sampath?
Yeah, doctor. So, doctor, I have a question on quality tests. So, for small molecules, it takes 5 to 7 days to get the assay values. So, how much the exact time does it take to get in large molecules?
Large molecules, it is in vitro or in vivo. So, it depends upon the time taken by the in vitro cell line growth or PD response in the animals, if it is in vivo. So, it depends, like if you see that field grafting in vitro results are available in 14 to 20 days time. In vivo erythropoietin bioassay takes almost 40 days because it is done on mice. So it depends upon the assay. But in small molecules, it is HPLC based and 5 days for your microbiology. So within 10 days, you can release the batch.
Thanks, sir.
Kaushik?
Yes, doctor. So my question is basically about the vaccines that you have told that there are DNA vaccines, RNA, and then mRNA vaccines and cell-based vaccines also. So my question is, why will somebody go to a process where the time is delayed for the production? From DNA, it will become RNA, then to protein, whereas the mRNA vaccine probably will directly make the vaccine or the something like that. So does it affect the efficacy, and why people choose different forms?
You see, what happens in an organization, any organization, with time, the organization develops the expertise in different domain area. For example, if you see that Bharat Biotech is having very good experience in attenuated vaccine platform. Bharat Biotech's any vaccine, if you will see, they are attenuated vaccines. Means they killed whole bacteria, which is moderated. Virus, not bacteria. Serum Institute has developed with time the vaccine development using this sera collected from the animals. The Zydus Cadila R&D has developed the expertise in developing the DNAs for many biosimilars. So based on the technology platform expertise available in the organization, these different vaccines has been developed. Suppose I do not have the expertise in mRNA based vaccine platform in my organization. I will not think of developing the infrastructure for that first and then developing the vaccine. Whatever infrastructure expertise is available with me, I will utilize that. So that is the reason different companies has started developing different platforms for the development of vaccines.
But does it results in the efficacy differently, or? You
see, it depends upon the clinical trials. when they are developing, for example, when the NIV has given the attenuated virus to Bharat Biotech, they have not given only single clone. They have given multiple clones. Then Bharat Biotech has selected the clone which was having the best expression level. And from that best expression level, they have scaled up their process. Now it depends upon the technology platform for upstream also and downstream also. But ultimately, every biologics has to meet the endpoint criteria set into the clinical protocols. Efficacy cannot be less. Only thing that the development time, development strategies, development infrastructure, upstream technologies available, downstream technologies available in the organization decides the strategy which vaccine we are going to develop. Ultimately, vaccine has to have a endpoint criteria which is written in your clinical trial. And that is the safety efficacy.
Thank you, Doctor.
Shashank? Shashank, you have any question?
Yes, sir. Yeah, actually, what I would like to understand is so far we were talking about human health, but there are many companies who manufacture these growth stimulating agents, or say, for animal vaccines, or say for plant diseases also, there are companies manufacture this kind of biological insecticide or say herbicide. So whether that falls into small molecule or large molecule, means the workflow remains same, or there also it is different, sir?
You see that animal products, vaccines, majorly animal vaccines, they fall under the category of biologics.
Okay.
Okay. Now you are talking about the plant. So that plant related any product is not vaccine, is not protein.
Okay.
They are either alkaloid or other category of chemicals. So plant-based biotechnology or genetic manipulation in the plant genes to increase the yield of the fruit or whatever it is, it is a different kind of science in the biotechnology, but not related to biologics.
Okay. Okay.
Pramod, you have any question?
Yes, sir. So basically, to understand small molecule and large molecule, contamination will be major concern in which process?
Contamination.
Yeah.
You see, let us first discuss the small molecule. So in small molecules, we have two kind of products. One is non-sterile products, other is sterile products. Non-sterile products are oral solid dosage forms, liquid forms, tablet, capsule, oral liquids. There we have a limit of presence of microorganisms in your product. Like you can release your tablets having 100 microgram, 100 microbes in 1 gram. Acceptable, but there should not be any pathogens. Means your product can have contamination of 100 microbes per gram, which are non-pathogenic. You can release the product because these non-pathogenic microorganisms in the tablet or capsule or oral liquid, they will not degrade your product also, and they will not harm the patient also. But if you go to the injectable product, which are sterile product, you cannot afford to have a single bacteria or contaminant in your product in small molecules. Now come to the biologics. I think कि आप सब लोग जब मिलते होगे ये biologics या biotechnology companies के लोगों से, you may be encountering that they are saying their DS is not sterile, low bioburden product. What does it mean? And they are using your 0.22 micron filter, disposable filter, for filtering their drug substance. Filtering from 0.22 micron means you are making your product sterile. What is the disadvantage? Why they are calling? Because if they will call their DS as a sterile product where no contaminant is allowed, your GMP requirements are very high. Compliances, validations are very high. That is why every biological manufacturer, they are claiming their drug substance or API as a low bioburden, but they are using disposable 0.2 micron filtration for their DS. Why they are doing this 0.2 micron filter? Biologics products are mainly proteins. If any of the bacteria or fungi is even a single colony is present in your DS, protein as a rich source of carbon and nitrogen will promote the growth of that single bacteria in the DS. And when the bacteria will grow, it will release many proteases and lipases, which will degrade your protein. That is why the claims made by the industry are low bioburden, but the actual practice is the drug substance is filtered through 0.2 micron filter so that there is not a single microorganism available in drug substance, and that is not contaminated. And if you come to the drug product in the biosimilar, the same principle of aseptic handling is required because they are injectable. That is why what happened when this industry has started growing in 98, 2000, initially people, the manufacturers, they have installed the DS facilities where the drug substance are manufactured only. And later on, to convert DS into DP, they have used their pharmaceutical industries filling lines to convert the DP. Because in DP, the same GMP is required, same process is required, the formulation, filling, visual observation, capping, then sealing, then labeling, the same process for which the industry is already expert. Only the molecule is different. There they are filling paracetamol injection. Now they are filling filgrastim injection. So contamination point of view, biologics industry is more prone and cautious because the nature of API or the active substance is protein, and which can be degraded by the enzymes produced by even a single bacteria available in the product.
Thank you, sir.
Anybody else? Shashank?
Right. What I suggest is let us have the questions in the chat box because we are running behind the agenda, I guess. So we will have a short break and we will again reconnect at 11:15, right?
Okay.
So
now
those who are having
now.
further questions, please put your questions in the chat box. We will have a separate Q&A session in the evening. I think that time, Doctor can.
Okay. By that time, I will collect all these questions, and then we will in the evening we will have. So, Prasanna.
Yeah.
Or Prasanna. Now it is 11:10.
Yeah.
Can we connect 11:25 here?
Yeah. Okay.
I am not going to log off anything.
Right.
I will just mute myself, and 11:25 we will start the next session. And that session is, I think, a process workflow of biologics that will be taken by Kevna. I will be there. I
Right,
will
sir.
be helping the her.
Right, sir.
Okay.
Sure, Doctor.
Okay. Okay, we'll meet here 11:25.
Right. Sure.
issue.
Hello everyone. So I will take this, the next part is biological process flow, and I'll go through the very simple and basic steps of how these biologics products are manufactured.
Devna, you can start your video.
Yes, sir.
Kevna, your voice is breaking. Sorry to interrupt you.
Still it is breaking?
Yes, there is some background noise along with your voice actually.
One. So now it's okay? Or still any voices coming?
Yeah, it's pretty decent now.
Yeah.
Okay, so shall I start?
Yes, please.
Okay. Okay, so I'll go through the basic process workflow of biologics, and let's start with what is biologics. A biologic drugs is a product is produced from the living organism or certain components of living organisms. Biological products are a diverse category of products and are generally large and complex molecules like protein. Biologic drugs include a wide variety of products derived from the human, animal, or microorganisms by using biotechnology. With the use of biotechnology, one can derive the biological drugs. The types of biological drugs includes vaccine, blood components, cells, allergen, genes, tissues, and recombinant proteins. These are the various kind of biological drugs available in the market. What is the application of biologics? So there are a wide variety of applications of biologics. Biological drugs can be used to treat the numerous diseases and its conditions, and are more and offering more advanced therapies. And some biological drugs are used for the treatment of Crohn's disease, ulcerative colitis, rheumatoid arthritis, and other autoimmune diseases. These biological drugs can be administered by injection or infusion. They are not available in the form of tablet or any other formulation because they are very sensitive and they get easily digested and inactivated if they given to the oral route or mouth. So therefore they are supplied as a powders for infusion or solutions for injections. Some delayed or reverse cause of immune-related conditions change the lives of the people with rare disease and have offered hope for many patients who previously had no effective treatment options for their condition. So this is the importance of the biological drugs. These are some similar biological drugs approved and marketed in India. As concept of similar biologics or similar biological drugs is already explained by Nishos sir. So these are the various products, its active ingredients, its therapeutic area, and at what date they launched in India, and which company is marketing these products. So these are the some companies which are engaged in the marketing of these products. For example, Actorize is used for the treatment of the anemia, cancer, and CKDs. Approved on the 6th January 2014, and it is company is the Cipla and Hetero. Bezalog, the active substance is insulin, and therapeutic area is diabetes. Launched in the year of 2009, and company is Biocon. Another well-known companies product is Cras, Darbepoetin alpha, and therapeutic area is again anemia, cancer, CKD, approved in the year of 2010, and companies Dr. Reddy's Laboratory. Ipofit or Ericine, the active substance is erythropoietin, and therapeutic area is again anemia, cancer, CKD, and it is manufactured by the company Intas Pharmaceutical, Ahmedabad. Exemtia, adalimumab, and it is used for the treatment of rheumatoid arthritis. In the year of 2014, it was launched and manufactured by Zydus Cadila. Some other companies, Intalfa by Intas Pharma, Maptas by Intas Pharma, Nupai Intas Pharma. So these are the various companies which are engaged in the manufacturing and marketing of some similar biologics products. Now we'll see how it manufactured. There are typical steps involved in the manufacturing of biologics. So first step in manufacturing of biologics is the desired gene isolation. The gene responsible for production of a specific protein. Then this gene inserted into the vector, this vector into the host cell, do the cell expression. Then the next step is cell culture of that cells which having this recombinant DNA. Then cell banks establishment and its characterization. Then next step is the protein production. After production of protein, it should be purified. So protein purification, then analysis of the product, then its formulation and storage and handling of that biologic products. Now, first step is the cloning. So before starting the cloning, we will go into the basic of central dogma, how the protein is produced. in our body. So central dogma is a process in which the genetic information will flow from the DNA to RNA to make the functional product protein. So for example, this piece of DNA is having this particular sequence of nucleotides. It will transcribe in the form of the mRNA, that is messenger RNA. From this messenger RNA, it will translate it in the form of the particular protein. For example, any kind of hormone or antibodies. So this is the principle of central dogma, that how the functional product protein is produced inside the body. So from DNA to RNA is called the transcription step, and from RNA to protein is called the translation. This RNA to protein step will occur outside the nucleus, and this DNA to RNA process will occur inside the nucleus. So after moving out of the nucleus, the RNA will go to the ribosomes, and where it forms the protein with the help of the amino acid. So based on this coding sequence, the amino acid will come together and join to form a long chain of protein. Now we'll go to what is cloning. Cloning in biotechnology refers to process used to create copies of DNA fragments. So anything which is copied from one DNA fragment is called cloning. It's called molecular cloning. Or cells, if they are formed from the single cell, is called cell cloning, or organisms. So cloning basically from the unicellular system or multicellular system. From unicellular system, bacteria, yeast, mammalian cell, or insect. And in multicellular systems, transgenic plant or transgenic animal. Transgenic plants and animal are those plant or animals which already having one or more DNA from the another species. So they are transgenic. So cloning that occurs naturally are in the vegetative production in the plant. For example, water hyacinth producing multiple copies of genetically identical plants through the apomixis of or potato tuber, which can if we plant one potato, it can grows into the many potatoes through its tuber. So it's a mode of asexual reproduction, and also called the vegetative reproduction in plants. Binary fission in case of bacteria. So one bacterial cells can divide into the two bacterial cells with this process of binary fission. Parthenogenesis in certain animals, in which the development of the embryo from unfertilized eggs will happen. So there is no mating of male and female gametes. Cloning that occurs by manipulation procedures or biotechnology is artificial cloning. So previously we seen is the natural cloning, and cloning that occurs by manipulation, fetus manipulation procedures or biotechnology with the help of the biotechnology is called artificial cloning. For example, molecular cloning, where copies of the specific gene fragments are produced. As we see in the central dogma, that specific gene sequences can transcripted in the form of RNA and then into the protein. So that particular gene sequence or our desired protein, if we want some specific kind of protein, then we have to know about the sequence on the DNA. So that kind of DNA fragment we have to design. So it's called molecular cloning. Cellular cloning, when single cell organism with its exact genetic content of the original cell are produced in the cell culture. So it is on the cell based. Organism cloning, where a multicellular clone is created generally with a somatic cell or nuclear transfer. For example, in human cloning, it will be a cloning of human cells using the somatic cell. For cloning, we need two starting ingredients. One is the DNA fragment and another is the plasmid vector or expression vector, in which the DNA fragment can be inserted. This DNA fragment is usually gene or the functional region from a living cells, or it could be artificial sequence with properties useful for a researcher. So this DNA fragment, we need a DNA fragment which contains the sequence of our protein interest in our protein which we require to manufacture, or it could be artificial sequence we can design to produce our desired protein. So recombinant DNA is a technology that uses various enzymes and enzymes to cut and paste together DNA sequence of interest. So DNA can be cut from one place and desired sequence can be inserted into the another into the vector which requires ligation. So various enzymes are used in that. This recombined DNA sequences can be placed into the vehicle called vector that carry the DNA into the suitable host cell where it can be copied or expressed. So once the DNA is cut and inserted into the another vector, this vector will help this DNA to transport into the suitable host cell, and where it can be used the environment of that cells and can be copied and can be expressed in the form of protein.
Ms. Kevna, can we go a little bit slow? I think team has
yes.
Some queries in between, so we can go a bit slower.
In which part they have query?
Yeah. So.
Okay. I'll read it.
With the overall content, I'm saying. Yeah.
Okay. So, is anything any doubt in the things till now? Shall I explain again?
No, I think you can go ahead, but I think you can go a little bit slow on the content side. So maybe if they have some questions, they can maybe raise their hands. Yeah.
So with the use of the recombinant DNA, we can have recombinant protein. These recombinant proteins are the protein encoded by the recombinant DNA that has been cloned into the expression vectors that supports expression of the gene and translation of the messenger RNA. So through this figure, we can easily understand what happens in cloning. So this is a yellow color, is a piece of DNA which having our gene of interest, and it will transformation of this into the competent E. coli with the gene of interest. This gene of interest, it will accommodated in the plasmid or vector and inserted into the bacterial cell. This isolation of the plasmid expressing this vector. So the cell having this particular type of particular piece of DNA will be isolated, and now these cells will be grow or scale up for the isolation of protein of interest. So now we'll having this cells in the quantity which can express our interest of protein. Okay, so this is called recombinant. The product will be the recombinant protein with the use of this recombinant DNA. So as you see, this blue part is a piece of DNA which is incorporated into the vector. This vector is transferred into the bacterial cell. Now this, with the use of this bacterial cell, this piece of DNA will form this, will produce this protein inside that bacterial cell. So let's see one video which can explain these things very clearly.
Isolation of a specific DNA fragment
Able to see my screen?
Yes.
Okay.
Yes, we could able to see your screen.
Okay.
And transfer of this fragment into a plasmid vector. As a part of a plasmid vector, the DNA fragment could be easily amplified, sequenced, stored for long periods of time, and used for gene expression and other functional studies. Two starting ingredients of molecular cloning are a plasmid vector and a DNA fragment that has to be inserted in it. The DNA fragment is usually a gene or other functional region from a living cell, or it could be an artificial sequence with properties useful for a researcher. Plasmid vector is a circular piece of DNA that could be easily amplified in E. coli, stored for long periods of time, and easily manipulated in a test tube. A typical plasmid vector contains an origin of replication that allows it to be replicated inside a bacterial cell, a selection marker, for example a beta-lactamase gene coding for ampicillin resistance, and a multiple cloning site which could be cleaved with several restriction enzymes such as BAMH1, ECAR1, or NCL1. In many vectors, the multiple cloning site is surrounded by sequences of promoter and terminator that guide expression of inserted genes after the vector is introduced inside a cell. To be used for molecular cloning, both vector and insert DNA are treated with restriction enzymes that cleave double-stranded DNA molecules, producing overhanging single-stranded nucleotide tails. After their ends have been prepared with restriction enzymes, vector and insert are combined together, and another enzyme called DNA ligase is added to the mix. And while complementary base pairing of single-stranded overhangs brings the ends of vector and insert together, the DNA ligase fuses them into one intact DNA molecule. In order to make multiple copies of this molecule, the ligation mix is introduced inside the E. coli cells in a process called transformation. During the transformation, the cell DNA mixture is kept on ice and then exposed to 42 degrees Celsius. A sudden change in temperature drives the DNA inside some of the E. coli cells. Then the cells are plated on a plate with growth medium, supplemented with a selective antibiotic. Only the cells that acquired the plasmid have resistance to the antibiotic and can grow on such a medium. After overnight incubation at 37 degrees, each transformed cell produces a colony of identical cells, oftentimes called a clone. The selected clones are then individually picked, grown even further in a liquid medium, and the DNA is extracted from them. So, in the process of molecular cloning, a DNA fragment that represented a tiny fraction of cell genome is integrated into a bacterial plasmid. As a part of the plasmid, this DNA fragment represents a quarter or more of total DNA in a test tube, and it could be effortlessly and endlessly amplified in E. coli. This presentation was brought to you by the authors.
Okay, so I hope this molecular cloning is clear to all of you. So now we can move to the cell bank. Okay. So in previous section, we just prepared a bulk of these cells, bacterial cells, having our having the piece of DNA which can give the our interest of protein. So this protein, the piece of DNA now can produce the protein that we require to produce it bulk. Now we have to preserve those cells once they are which are already engineered. So now we need to preserve those cells for the further production and for backup also. So the next step is the preparation of the cell bank. So cell bank is a facility that stores the cell of the specific genome for the purpose of the future use in a product or medicine needs. So creating a cell bank is a vital element in producing consistent biological products. So for biological products, this cell bank is a kind of raw material. So it needs to be handled with a great care. Cell banking systems are set up to assure a uniform population of a cell is preserved, and this integrity should be maintained. That it should not be contaminated, as they are the bacterial cells. So there are the chances of contamination with the other organism. So it should be preserved and stored and maintained, handled in such a way that it should not be contaminated with the other organisms. And a sufficient supply of the material is accessible for the life of the product. So first is the master cell bank. Is derived and extended from a one parent cell culture, as we seen in previous section, how the cells are designed and cells is engineered for our desired DNA. So it is a master cell bank, and it should be expanded from it was expanded from a one parent cell culture, and should be dispensed. Now it should be stored. So for that, it should be dispensed in the various multiple containers or called cryovials, and stored under the defined conditions. This MCB is produced using standardized laboratory practices and procedures, and characterized depend on the strain to ensure the proper identification to demonstrate the cell bank is free from the accidental contamination. That means MCB should, once we have this MCB, it should be properly identified, and it should be demonstrated that the identification should be done on this MCB, that the cells are desired cells and it is free from any accidental contamination. So the culture is very pure, and that has to be now stored. Now from master cell bank, there will be a working cell bank. So MCB is used to derive all working cell banks. Now these working cell banks will be used for the production purpose, and MCBs will be stored as a backup of that pure cells. So the working cell banks is prepared from one or more MCBs vials, and similarly the MCB before being used in the production. Usually WCB is produced from the one single vial of MCB that has been characterized for its viability and contamination. These working cell banks are generally used for the next stages of the manufacturing. So this is a picture of MCB versus WCB. This MCB is produced from our originally engineered cells, and this WCB is.
Hello, Khivna. This is
Yes.
Piyush. So can you explain the earlier slide, more clearly, so that everybody can understand, because that's a very important slide.
Which one? This
Yes,
slide?
yes.
Okay. So MCB is a original culture which already designed we already designed in the previous section. So it's called MCB, master cell bank. So its purpose is to produce a WCB, and for the storage for the future use in case this WCB is contaminated or anything viability is lost. So we have this MCB we have to preserve this MCB for a future use. In case of WCB will not work. So in that case, we again reconstitute this MCB and produce new WCB. Okay, so this MCB is used to derive all working cell banks. So in production purpose, we'll use this WCB, not the MCB. So working cell banks are necessary for the later stage of the development and manufacturing of the products.
So are we saying here that the overall cell bank also has several categories, right? One is the master cell bank, where we will keep pure cells, and then we will be having a working cell bank.
Working cell banks, which is also the same as the MCB, but which is derived from the MCB, which are same as MCB. But this WCB will use for the production purpose, and MCB will use for the use as a preserved cell bank for future use. If any case where the WCB will not work, is not working, or it is contaminated, so we have a backup plan as a MCB.
Okay, 1 minute. Let me explain this process. Initially, what happened? There is a question on the chat box also. I have seen where these activities are done. So this cloning, like our DNA fragment and the vector and then the insertion in the cell, that all is done in R&D lab.
Laboratory.
Or R&D lab. And once we get the expression, what we have seen in the plate, that only the ampicillin resistant cells will grow on plate. These cells are called, the one cell which we have taken from the plate, this is called research cell bank, which has been developed by the R&D department. Now we have only 1 or 2 cells. They have been grown to prepare large number of tubes like this, which are called master cell bank. Because this cloning and the insertion and all these things is done once, and the best expressing cell is selected by the R&D lab. And then R&D cell bank is converted into master cell bank, which is stored for lifetime. Because reason is, if once we get the marketing authorization from one master cell bank, we cannot change the master cell bank for lifetime. That is why this preparation of master cell bank is very important, because we don't know how many years, 20 years, 30 years, we are going to manufacture this product and sell this product. Now for routine use, we cannot use this everyday master cell bank because they will be exhausted. We can produce 200, 500 master cell banks. From master cell bank, we prepare the WCB, like here, one cell line, this is R&D cell line, and then culture, and then the master cell bank of hundreds, it is stored in the nitrogen container. From this one of the master cell bank, we have prepared working cell bank, which is used for day-to-day production. Why this is? Because master cell bank is a repository. If for any reason, any point of time of the life cycle of product, my cell bank is contaminated or not working properly, I can go back to master cell bank, prepare a fresh working cell bank, and start using for the production. That is why these two things are done. Now, Sagar, you have some question. Sagar.
Yes, my question is a little bit relative to our, to the chemical part. So generally, when it comes to chemical, we have primary standard and we have working standard. Is the same concept what I see?
No. Primary standard and working standard are used for the analysis purpose.
Okay.
But here this MCB and WCB are your raw materials for manufacturing your product.
So in this case, a master cell culture is used as a reference for the purity check for WCB?
No. Master cell bank is a copy of WCB. Master cell bank is prepared in hundreds of numbers for a lifetime repository.
Okay.
And WCB is working cell bank. From this WCB bank, every batch is manufactured using one vial.
So connecting to same one question, doctor. When is as it is used as a depository, right? Does it change their properties?
No, that is what tested in the characterization of WCB and MCB both. Their stability is also done for how long they are stable. That is why they are stored either at liquid nitrogen or carbon dioxide.
Okay.
They are permanently stable cell lines. MCB is master cell bank, which are derived from your research cell line, where this is developed. And WCB is prepared for routine use from the MCB.
Okay, got your point. Thanks.
Both are equal, characterized, and stable.
Yes, I got it. If
But
something
why these two cell banks are made? WCB container you will be opening day to day, every batch for every batch. A lot of handling is happening.
Yes.
So sometimes something can go wrong and you may lose or you may damage your WCB. So you have a backup to reconstruct your WCB from your MCB bank. Okay, Shekhar, you have any question?
Yes, doctor, as you said just now that for WCB, for one vial, we are making one batch. Similarly, from one MCB, how many WCB you can make?
You can make any number. You have to decide based on your production and all these things. This is the individual company's call. How many? What is their projection for next 10 years or 5 years or 20 years?
Okay. And both of them should be stored in a liquid nitrogen?
Yes, they are stored in a liquid nitrogen.
Okay. And what will be sample size? Like it will be in 1 ml or less than 1 ml?
They are generally 1 ml in cryo vials of 2 ml.
Right. Okay. Thank you.
Prasanna, you have any question?
So there is one question from Mr. Subhash Lal that patent is given to MCB or RCB?
Patent is not given to any live organism. Patent is given to only processes.
I have a question, Doctor. In this cloning process, as well as MCB and WCB, is there any fluid transfer application associated where a peristaltic pump or any kind of device is used?
No, in this all, because they are in very small quantities, only the micropipettes, disposable tips are used.
Okay.
Once it will go to fermenter, then your transfer devices will be required.
Okay. And this making this MCB is a repetitive process, right?
No, making working WCB is a repetitive process. MCB is prepared once.
Okay.
And its characterization, stability, everything is included in the marketing authorization application. Plus these MCBs you can store at your facility or at a remote location. And there are provisions on the agencies located across the world like AACT. You can deposit your MCB for lifetime at their custody, so that any time in future, if you find something wrong in your working cell bank, you can go back to your master cell bank and retrieve that.
Okay.
Dr. Saxena, Piyush here. So
yes,
if we assume that RCB determines the capability of an biosimilar organization to develop new products in future, will that be right?
Yes, definitely, because for every product, the R&D has to develop a RCB, research cell bank, where all these vector and fragment, DNA fragment and the host cell transformation, all these activities are required. Then the expression level, based on the expression level and the quality of protein, the RCB is identified.
Okay. So when we decide for a new product and we decide on a cell line, then we go for the MCB. Is that right?
When we decide a product, my next session is based on that only. Let me explain briefly.
Okay, fine.
Next session will be giving you the correct idea how this development is done.
Perfect. Sorry.
Because this is a complete process flow from cell bank to the fill finish.
Fine, perfect. Thank you.
Damalia Basu, you have any question?
Yes, sir. I have one question that this process is under upstream.
This process is molecular cloning process, not an upstream. Upstream will start once you will inoculate your working cell bank into a flask or in a fermenter.
Okay.
Okay, and there are few questions. Chat box questions we will have at the end. Can we say that these are basic process for even stem cell banking? Yes, definitely. Deepa has asked this question. When we develop the stem cells are derived from the human body. And then we do the cloning of that stem cell. They are also stored like this only, that small quantity, millimolar quantities in Eppendorf cryo vials. The same cell banking is used for the stem cells also. can we say WCB is diluted version? No. WCB is as good as master cell bank. The nomenclature difference is only working cell bank is used every day for the manufacturing of your batches. Master cell bank is a repository or a backup for your working cell bank. Any other question? Shekhar, you have any question? Okay, Kevna, you can move ahead.
So after completion of this MCB and WBC, after having enough WCB vials, we'll move to the upstream processing. So for our production and this process will start from the upstream processing. So upstream processing is the first step in which the biomolecules are grown, usually by the bacterial or mammalian cell lines in bioreactors. There are bioreactors in which we'll use this WCB for the growth of bacterial or mammalian cells. When these cells reach at the desired density, or when these cells will multiply into that medium and will reach at the desired density, they will harvest and move to the downstream section of the bioprocess. So initial steps of the upstream process will start with the milliliter quantities of these cells, which is working cell banks. These working cell bank vials will be inoculated into the specific media meant for them, designed for them, and these cells will grow inside that medium. And now this medium, which having some desired quantity of cells, will use into the production to produce a specific protein molecule. So it's called cultivation step in a control environment like temperature and pH, pressure. This environment, the favorable environment should be provided to this cells. So the cell culture to grow into the volume to the several thousand liters to produce the several kilograms of API, desired protein products or API. So we'll start with this milliliter quantities of the cells, which will grow into the several kilograms or several thousand liters, and we'll get the which will produce the several kilograms of the desired API. Mammalian cell culture is used to produce, generally used to produce a large and complex protein molecules such as monoclonal antibodies and microbial organisms, for example, E. coli culture, which grow much faster and with the cultivation times of several days and several hours. The doubling time is very less, 20 minutes. So they can grow very fast in several days or in several hours. We'll have a large population of cells. So small size molecules such as peptides, enzymes, antibodies, protein scaffolds can be produced very efficiently with the microbial cell lines. The first step for microbial for upstream processing is the media preparation for the growth of the cells, which are already which cultivated into the laboratory into the flask. So it is transferred to the high quantity into the bioreactor. So it requires some specific media. So some specific formula required for each bioreactor stage of scale up of the inoculum. This media preparation is generally done into the tanks or carbos or bottles or bags. So this media is having specific nutrition. Media is nothing but it's a nutrition content of the required for this bacterial cells to grow. So similar to human, the cells also require some proper nutrition to function in a proper way, and ultimately to produce the protein products. So they require some specific nutrients. So media are designed in such a way that it contain all the nutrients which is required by the bacterial cells or mammalian cells. Therefore, the media is predominantly made up of the various components like carbohydrate, nitrogen, fats, and the trace amount of salts. So this is the media ingredients. Now media preparation. Now all this this media is in the form of the powder. This powder will dissolve into the water for injection and produce a homogeneous mixture of content. This mixed solution now transported with the help of the bag or carbo to the bioreactor using a pump. The required accessories are assemblies, tubing, bioprocess bags, filters, pumps, plastic fittings and connectors, instrumentation, bottles and flasks to transport this media from its container from the site where it is prepared to the bioreactor tanks or fermenters. The next step is the cell culture. Now we have a quantity of cells. We have a media. So now the next step is the cell culturing to grow these cells into the media. Cell culture is the growth of the cells outside the body. It's called in vitro. So this is the in vitro growth of the cells. All living cells grow by the cellular division, and same applies to the cell culture. So in cell culture stage, these cells will divide into the specific density. And they will multiply once they get a suitable environment inside the bioreactor. For example, pH or temperature or pressure. The correct environment is the growth medium as a food source and the cell culture vessels for controlling gases and temperature. So a bioreactor is designed in such a way that it can control the gases and temperature produced during at the time of growth of the bacteria or cells. So as the cells grows inside the medium in bioreactor, it will turn into the opaque color as the cell density increases. As the cell multiplies inside the medium, inside the bioreactor, the liquid will become, will turn into the opaque due to the growth of the cells. Now this growth rate is regularly, every day or periodically monitored to determine how the cells are growing over particular cycles. So there is a particular cell cycle of each and every cell. There is lag phase, log phase, stationary phase, and decline phase. So ones need to monitor at which stage the cell is growing. So it is carried out by taking periodic samples from that bioreactor and precisely counting of the cells. So when we reach that particular density of the cell, we have to turn off that process. So every day it is need to check the samples from the bioreactor, and the required accessories are tubing, bags, filters, filter pumps, plastic fittings and connectors, plastic equipment, instrumentation, bottles and flasks. Now up to the cell culture, it is upstream processing where cells grows to the desired quantity, where cells are having at desired density, which we require to produce to get our product in.
So let us have a brief Q&A here. One question, two questions are there. Are this cloning process same for both intracellular and intracellular protein of interest? Yes, this cloning process is same for intracellular or intercellular proteins. The second question is, where is drums and liners used? Drums and liners are not used in fermentation technology. Drums and liners are used in the storage of raw materials, mostly in the fermentation, mostly in the pharma industry. Biopharma industry, drums and liners are not used. In upstream, Anybody is having any questions?
This type of media, pH and temperature are decided in R&D stage. Yes.
So no questions, then you can move.
Sir, there is one question. The type of media, pH and temperature are decided in R&D stage. It is decided at the time of the product development stage.
Yes, that
R&D.
the media composition and the temperature, pH, oxygen demand, RPM, everything is decided at product development stage.
Dr. Saxena, the decision on the excipient side has been done during this stage only, or in some other stages?
No, there are two different things. One is raw material, other is excipients.
Yeah.
So let us compare this with the small molecule. So the clarity will be very good. So raw materials are required in both small molecules and large molecules. The raw materials means the materials which are used to manufacture the product. And we can call it, the product is active pharmaceutical ingredient, which in biopharmaceutical industry, we call it drug substance, or ICH terminology for the API, active pharmaceutical ingredient, is drug substance. So all raw materials which are going to manufacture the API or drug substance are called raw materials. Now, once we have our API, that is drug substance, we need to prepare the finished product, which is called drug product. So simply, API will not be stable or will not be give the desired physiological function. For that, we need to add certain ingredients like surfactant, stabilizers, and to make the finished product stable and functional. These ingredients are called excipients. Excipients are directly entered into the human body, where raw materials are not going, even the traces of raw materials are not going into the human body. This is what the difference between raw materials and excipients.
Doctor, excipients are added in formulation area or
yes,
in the upstream?
When we do formulation using our API, adding a buffer solution, in that we have added polysorbate 80, that the buffer component and polysorbate 80 are excipients. Polysorbate 80 is a surfactant which is used to disperse the protein molecules so that they should not get aggregated.
Yeah, okay, fine.
What is the frequency of cell growth monitoring check? 1 minute. What is the frequency of cell growth monitoring check? Okay. Like in the slides you have seen, there are two kinds of cell cultures. One is mammalian cell culture, other is bacterial or yeast cell culture. It depends upon the doubling time. Doubling time means in how many minutes or how much time the n number of cells has become 2n, double the cell. So in bacterial cell culture, the doubling time is very low. Approximately for E. coli, you say it is 20 minutes. So every 20 minutes, the cells are getting doubled. So when the doubling time is very low, the frequency of monitoring of growth is very less. For example, it can be hourly, 2 hourly, 3 hourly. Where the doubling time or the doubling time of the cells, like in mammalian cell, CHO cell line, Vero cell line, CHO cell line is better than the Vero. Where the doubling time is around 48 hours, 72 hours, even Vero cell lines, it is 10 days. There the frequency is very high. You can monitor every day or every alternate day. It depends upon the growth rate of the cells. So somebody was asking one question. There is a question how contamination or growth of organism is removed from preparation of media buffer added to bioreactors. Yes. The removal of contamination, because we know that powdered media is non-sterile media and the salts used for the preparation of buffer, they are non-sterile. The containers in which they are prepared, they are non-sterile. So first the media or the buffers are prepared to a desired strength in a container, and then based on their stability to the thermal cycles, either they are sterilized, or if there is any component which is thermolabile, they are sterilized with filtration before inoculating into the fermenter. Any other question?
Doctor, in mammalian cell method, suppose the doubling is taking few hours or more, why that is preferred?
Pardon, can you repeat your question?
Yeah, my question is like, compared to bacterial and cell growth, the mammalian cell growth you said takes 48 hours or more instead of a few minutes, 15-20 minutes in the bacterial cell doubling.
Right.
So then why it is preferred mammalian cell doubling? Where it is preferred? In what condition?
You see, mammalian cells and microbial cells both are grown in fermenters only, bioreactors. Generally what happens ki microbial cells grown in the free state in the bioreactor, they are suspended in the liquid medium. But mammalian cells are not grown in suspended condition. They need a surface where they can stick and they can multiply. So if the fermenter, single-use bags are majorly used in mammalian cell culture, where the bag's inner surface becomes a surface to adherent of the cell lines where they are growing. And why there is a difference in the growth rate in microbial and mammalian? The same reason microbial cells are present in the suspended form of liquid media. What is happening when we are doing fermentation of bacterial cell line? Generally, we are sparging the air. Why air is required? Air is required for the growth of bacterial cell line. And these bacterial cell line, from the air bubbles present in the media, they take oxygen for the growth. In contrast to this, when we go to the mammalian cell culture grow on the availability of carbon dioxide, which is a source of carbon for them. So mammalian cell culture is done in the CO2 incubators, while the CO2 is purged in the disposable bioreactor, while the bacterial cell culture is done in the bioreactor where air is sparged. That is why the growth of bacterial cells are much faster than the animal cell lines. Any other question? Can we move? Yes, Prasanna.
So the accessories or products, what you have mentioned, which are used in this process. So is it recommended that these accessories are pre-sterile, or is it okay if the customer uses autoclaved products?
You see, when there is a single-use technology used, everything should be pre-sterilized.
Right. Okay.
And before going to fermenter, before going inside the bioreactor, either you can use pre-sterilized single-use connectors, tubings, bags, or if somebody is wanted to recycle these things using glass bottles or SS containers, they have to autoclave before entering into the bioreactor.
Right. Okay. Okay.
Any other question? So, Kevna, you can move to downstream now.
Okay. So as we've seen, the growth or production of cells is up to the upstream processing. Now the desired protein product is already produced as the cell is grown up to this particular density. And so now it is time to harvest our product. So it's called downstream processing, where harvesting and purification of the product is done. So first is the cell harvest. So first step is a cell separation from, for the first step, the first step is a cell separation from the culture. Timing of the termination of the cell culture is crucial and predetermined based on the quality and quantity of the product accumulated in the bioreactor. So it is predetermined that at this time, at this cell concentration, at this cell density, we'll get the desired quality and quantity of our product. So at that time, it is need to terminate the process in the bioreactor. The first step in the mammalian separation is usually a centrifugation in the technique to separate the cells from the culture through the rapid spinning and the sedimentation. So the cells will be separated out from this other media. Microbial harvest is similar to the mammalian, however, the lysing step must be occur prior to the centrifugation. As we seen, this bacteria can produce the protein product internally. So to get this, our desired product, it is need to lyse the cell. So after lysing of cell, protein product will liberate into the media, and that we need to recover. So for microbial harvest, it is required to lyse the cell for the recovery of our product. The next step is the filtration to remove the large debris through the depth filtration. So first is the large debris of the media residues and other excipient residues and cell bodies. We need to separate it from our product. So this two-step will increase the transparency of the mixture. As we see in this culture, the media will be opaque at the growth stage. So when we centrifuge and filter our media, so the large debris will be removed from this media and the product will get more clear than the previous one. It's also known as the clarification step. Okay, sterile grade membrane filtration using 0.22 micron filter or similar filter is often used to harvest or remove the small particles and microbial contamination from the during harvesting procedures. So sterile grade filtration is also applied to separate our cells from the product. The required accessories are assemblies, tubing, bioprocess bags, sampling system, filters, pumps, plastic fittings and connectors, plastic equipment, instrument, bottles, and flasks. Now the next step is purification. First, we have harvest our product and it is separated from this cell debris. Yes.
Kevna, can you please move back to the previous slide once again?
This one?
Yeah. All right.
So Kevna, what is the filtration requirement? It is the only 0.2 micron, or we should have some kind of TFF clarification and
it will come into the next step. This TFF and NFF will come into the next step. This is a preliminary step. Preliminary, we have to separate our product from the debris. So this is a very initial step to separate our product from other debris and other contaminations, other things.
Okay.
Big, large particles. Yes.
let me explain in a little more clarity. We have two kind of fermentation. One is microbial fermentation, other is cell culture based fermentation. So cell culture based fermentation, always the product is manufactured outside the cell. It is extracellular. So whatever protein is manufactured by the mammalian cell line, it will come into the media inside the bioreactor. So in mammalian cell line, the clarification or filtration is only the separation of the cells from the rest of the media, which contains media ingredient plus our product. In bacterial cell culture or fungal cell culture, there are two kinds of products manufactured. One is intracellular or other is the extracellular. So extracellular process is same as cell culture because the product is liberated out of the cells. But most of the E. coli products, where E. coli is used as a host cell, the products are manufactured, stored inside the cell. There is no product available outside the cell in the medium. So what happens if we want to get that product from inside the cell, which is closed, before this centrifugation, what we need to do, we need to break these cells. That is what we call lysis of the cell. So that the product is out of the cell, and when we centrifuge or filter, the cell, broken cell or cell debris are separated out from the media, including the product. So this is what the first step when the product is manufactured inside the bioreactor. Now you can continue. Prasanna, you have any question?
No, sir. Sorry. Yeah.
And there are two questions on the three questions on the chat box. Let us clear that. Why we used two terminologies like fermenter and bioreactor? Very good question. Actually, when this biotechnology has started, it is very long back. It is as good as the pharma industry. Initially, this technology was used to manufacture the alcohol or the yeast. So that time, that is called the fermentation. That process is called fermentation. Like our making of curd is a fermentation, or vinegar, manufacturing of vinegar is a fermentation. So from that fermentation, classical fermentation, that terminology has come as a fermenter. Now in modern days, since in pharma industry, we carry out reactions in the reactors, we have started calling this fermenter as a bioreactor where the manufacturing of the product is happening. So this is what the difference between fermenter and bioreactor. Now next question is role of cell separation and harvesting in upstream. Cell separation and harvesting is done to isolate or separate our product from the entire media, which is what we call it mother liquor, entire media, water, and the all cells. So we are doing harvesting and cell separation to isolate crude product from the entire bioreactor.
But Doctor, my query was that, so when we start with the inoculum development, way from media preparation to till cell separation, as and when it reach the desired density, they will be harvested. What is my understanding is that all the process which is related to upstream processing or which comes in downstream? Because as and when ma'am started this cell harvest, cell separations and harvesting, which downstream processing. So I just want a clarification on,
So cell separation and harvesting is downstream processing. It is not upstream processing.
But inoculum development, which comes from media, I mean, it starts from media preparation to harvesting, no, doctor?
No.
Then it has to be-
Inoculation development to achieving the desired cell density comes to the upstream.
Yeah.
And harvesting is come into the downstream.
Yeah, but in this cell separation, comes in the previous slide, which we saw, no? That I have seen, which comes in downstream processing. So that's where my...
Okay. So that may be a mistake in the slide.
Yeah,
but
yeah,
yes, basic fundamental is up to reaching to the desired level of cell density or product concentration in the bioreactor.
Yeah.
Is falling under upstream.
Upstream, absolutely.
Any material taken out of the bioreactor for further processing is called
harvesting. Harvesting, yeah.
That comes into the downstreaming. So we will see. I have one module where I will show different kind of fermentation. There you can see that upstream and downstream can go parallelly also.
Okay.
So next question is, what are flows and pressures involved in cell harvesting? You see, it depends upon many factors, the design of plant also, and the capacity of your downstream equipment. For example, your centrifuges. Earlier days, if you see, the plants are built horizontally. Then one room is adjacent to the other room. When we are designing the plants horizontally on single floor, for harvesting, you need a device which is called pump to transfer the liquid from one place to other place. And the flow depends upon your receiving end capacities. So flows are decided based on your capacity of centrifuges. And pressures are designed based on the desired flow rates. Nowadays, what is happening? The plant design are happening as a vertical. One room above the above room. So the transfer of material is done through the gravity pressures. And the flow rates are controlled by the parasitic pumps. Any other question? Okay, you can continue.
Okay. The next step is purification after harvesting of cells. Is the most important step to separate and purify the molecules from the complex feed stream. So biological products are very effectively purified by chromatography. Most of the biological products are protein and they are effectively purified by the chromatography. So chromatography is a technique for getting high purity products, especially with the proteins. So there are many methods of executing the chromatography step. These are the ion exchange chromatography. These are the different chromatographic techniques to purify the proteins. Hydrophobic interaction chromatography, affinity chromatography, and gel filtration chromatography. So these are the various different principles of using this chromatography. Gel filtration is also called the size exclusion chromatography, which separate the material on the basis of size and shape. Ion exchange, it separate the material based on the net charges. Chromatofocusing is also working on net charges. Affinity is working on the principle of biological affinity. Hydrophobic interaction is working on the polarity of the substance, that is hydrophobicity of the molecule. and immobilized metal ion affinity, working on the metal ion binding. So these are the various chromatographic techniques working on the different principle and using of this chromatography based on our desired protein. So typical biopharmaceutical process uses 3 chromatographic column steps, and each preceded by the UF/DF of the product, that is ultrafiltration and diafiltration of the product, is done before the product enters into the specific columns. So it is necessary to prepare the product for the following chromatography stage because all columns are operating under different pH and molarity conditions. So it is required to prepare the product before it is entering into the specific chromatographic column. So UF/DF is performed, which concentrate and resuspends the product into the correct buffer before it is placed into the column. So basically the size is the basis of the separation of the filtration in most cases of UF/DF. So under application of this pressure force, the membrane will retain the larger molecules such as protein, and ultrafiltration operations can be performed in the tangential flow filtration or with the normal flow filtration. In this case, the protein, our product, which is a large molecule, will retain onto the membrane, and other media and other things will comes out, and it will be removed as a filtrate, and our desired product will come as a retention. So required accessories are assemblies, tubing, bags, sampling system, filters, pumps, plastic fittings, connectors, valve, plastic equipments, and instruments. Now the next step is the concentrated DS. The filtrate is free from the suspended particles and usually contain 80 to 98% of the water. The desired product is a very minor constituent as it will retain onto the filter, and it has very minor constituent, and the water has to be removed to achieve the product concentration. To get the desired product concentration, or is called bulk drug substance, we need to remove the water from that content. So it can be done by using evaporation or liquid extraction or membrane filtration or precipitation and adsorption. We can concentrate our drug substance to its desired concentration. So actual procedure is adapted based on to the nature of the product and the cost factor. Now up to the drug substance. Getting the drug substance is the downstream processing.
There is a question on this part. Okay. Can we combine multiple chromatographic technique to purify one product? Yes, this is always a combination of technique. At least 3 different types of chromatography is to be performed for purification of the product. That depends upon what is the kind of impurities are there in your product. So majorly, first step is done by the ion exchange chromatography, then gel filtration chromatography, and then last one is your polar and non-polar chromatography.
That's why it is required to prepare our product before it is entering into the chromatographic columns.
Yes, TFF and NFF is tangential flow and normal flow. Tangential flow and normal flow difference is the surface area available for the adsorption. Okay, you can continue.
Now, process flow, workflow for the field finish. First is aseptic processing. Aseptic processing is a step-by-step method of making formulations, that is drug product, which will fill into the vial, syringe, ampule, or cartridge. And the aim is to get the safe, effective products are free from the microbial or any other contamination. So priority step involves in the aseptic processing is the thermal sterilization of the product, sterilization of the packing material, and the conservation of the sterility during the packaging. So thermal sterilization, if the product is heat stable, then can go for the sterilization, thermal sterilization. If the product is heat labile, then it can be filtration can be done with the product, and the final product is filled into the vial, pre-sterilized vial or ampule. And the sterility should be intact during the packaging. So the conditions should be maintained during the filling of the drug product into the final its primary packaging. After bulk drug substance arrived at the manufacturing facility, it should be stored into the very controlled condition until the the filling date is scheduled. So safety measures should be monitored every 24 hours. The safety measures include temperature monitoring and this humidity monitoring and all that, and it should be stored under very controlled condition. So ideal step of thawing steps are performed in closed system to prevent the exposure of BDS to the potential microbial contamination. So before using this drug substance for the drug product, it should be properly thawed. So this thawing step should be performed in the closed condition to avoid the microbial contamination of the drug substance. The next is the pooling and the formulation of the final drug product that takes place into the compounding and formulation chute in either small or large tank under the controlled condition. So to get the desired concentration of drug product, the drug substance to be properly thawed and formulated as per our desired concentration. Now this drug substance will be called as drug product. Proteins are highly susceptible to the loss of the biological activity, hence the formulation requires the special care. Certain stabilizing additives are added to increase the shelf life or stability of the products, such as sugar, salt, polymers, or glycerols. These materials can be added at the time of making drug product from the drug substance for its stability. So proteins are fermented in the form of solution, suspension, or the dry powder. All equipment and components must be sterile and free of pyrogens. So these are the various preparations can used in the packaging and filling of the drug product. Tools going into the filling lines and isolators should be sterile. Vial preparations should be sterile. Syringe preparations should also be sterile. Stopper and cream cap preparation all should be sterile and free of pyrogens. There are several systems for the sterilization of equipment. It's autoclaving or dry sterilization, fumigation, radiation, and E-beam for the sterilization of equipment. There are some questions.
Okay, let me go to the chat box. There are many questions. Okay, the first question is, why some customer prefer to use centrifugation step than TFF step, which is considered the best method of more yield? You see, it depends upon the product, because in TFF, the losses are much higher, but the chances of getting contamination is less. So it depends upon the process and the product, which method is used for the separation of or the concentration of the product. And centrifugation is the simplest method, but not a sterile method. TFF can be a sterile method. The next question is protein purification. In which condition dialysis membrane or cassettes can be used? Dialysis or cassettes are used generally as a replacement of ultrafiltration or diafiltration for the concentration of the protein before loading to the columns. The next question is, can you please explain the scale-up concept with respect to upstream and downstream? Yes, Prasanna, we have a separate module for that.
Sure, sir. Okay.
Okay. After filling, it is a sterile product or non-contaminated product, how it is done? After filling is done from the formulation tank. We transfer the formulated material to a filling tank. When this transfer is done through the pipeline, disposable pipeline and disposable 0.2 micron filters, the product, final filling product is sterile because it is filtered through 0.2 micron filters. When it is going from the filling tank to the vial or the pre-filled syringes, again another 0.2 micron filter is attached. So the material 1 ml or 0.5 ml, whatever fill volume is going into the vial, it is 0.2 micron filtered multiple times. So it is a sterile product, and vials and bunks and the caps, they are also separately sterilized through tunnels or the autoclaves. So ultimately your final product, using your product sterilized with 0.2 micron filters, and your primary containers like vials, rubber bunks and caps, they are autoclaved or DHS, dry heat sterilized in the tunnel before filling. Some of the products which are not able to be filtered through 0.2 micron, for example, viscous products, they are sterilized by terminal sterilization, that is called after filling the product into the vial or primary container, they are autoclaved in a autoclave. So your final product is always a sterile product or non-contaminated product. E-beam is a method of energy rays which are thrown on the containers of the product, and they kill the bacteria inside the product. Is this stage to decide pharma inactive ingredient like buffer excipient? Yes, this is the stage when we are adding bulk to the formulation tank along with buffer and excipient. Piyush, you are very correct. This is the stage where we have to decide and finalize the buffers and excipients.
Thanks, doctor. Thanks a lot.
Because you have to just, only one thing you have to remember, that what is the difference between excipients and the raw material. Raw material will never go inside the body, and excipients are going to be 100% they are going inside the body. That is the major difference. What are the types of bioreactors used in bioprocessing, majorly preferred in industry? The bioreactors, they have started, the industry can use the glass reactors, industry is using the SS reactors, and now gradually all these glass and SS bioreactors are getting replaced by single-use bioreactors. And we all know that now up to 20,000 liters single-use bioreactors are available, which are fitted with your exhaust gas nozzle and the inlet nozzle, even the Stirrer also, magnetic drives are also attached with this bioreactors. During this mechanical transaction with cell in downstream, what are expected loss percentage? Are there any other ways to minimize? You see, it depends upon the process, the company organization, how they have optimized their processes to reduce the losses. In some cases, the losses are up to 20% to 80%. Next is every stage of manufacturing microbial testing is done. Yes, every stage of manufacturing microbial testing is done to ensure that my process, any of the unit operation is not contaminated. You take sample, send it to microbiology for any contamination. Filters mean capsule filters or syringe filters. Capsule filters are used for the filtration of bulk solutions before the final filtration. And syringe filters are used. Syringe filters are generally used in the lab, but the 0.2 micron filters, single-use filters are used in the aseptic processing. In which step 3D mixing bags are used? Generally, the 3D mixing bags are used in the bioreactor. What should be optimum loss accepted as per industry? It depends. Losses are calculated at unit operations. At every stage, losses are calculated and defined as per the process performance in the BMR. How can we expect bacteria as already we have used 2M? What is, I am not able to understand.
0.2 micron filters.
How can we expect bacteria as already we have used 0.2 micron? Where in at what stage you are expecting? What stage you are expecting for bacteria?
Filling.
So we cannot, that is why we are using 0.2 micron filtration to remove any bacteria.
Okay, yeah.
Yield of product is dependent on which stage. Yield is calculated, as I told you, yield is calculated starting from your harvesting procedure. And at each stage, you have to define the range of yield at every stage. So it is not fixed. It may happen that in the previous stage, your rejection is 10%, but in subsequent step, you are losing product by 50%. Because as the purification increases, the losses are higher. Any other question?
Okay, so shall we move to the filling stage?
Yes, you can move.
Yes. The solution in suspensions, accurate filling methodologies prevent underfills, which causes the rejects, or overfill, which increases the cost. So the filling should be in the proper volume to prevent the underfills or overfills. So every at the time of the filling, the filled volume shall be checked. So there are various filling types, which includes pressure fills or peristaltic pumps and diaphragm pumps for filling of the products. Biology and weight variation. Achieving high yield is absolutely critical in biologic products because they are extremely high value. So to ensure that every container is properly filled, it is required to check the weight of the product which is filled. So there should be accurate in-process measurement of the volume within the containers. So we can check that the product is properly filled into the container. It is not overfilled or underfilled. Next step is lyophilization. In case of the powdered product, the lyophilization step is required. It is a freeze drying used to stabilize the formulation of the sensitive parenteral drugs, especially biologics. To provide the more stability to our products, the lyophilization technique is required. The liquid is filled into the vials, which are then loaded into the lyophilizer, which is an instrument. The temperature is lowered into the lyophilizer, and the liquid freezes into the solid. So temperature is so low that the solid product, the liquid product will freezes into the solid. Then vacuum is created into the chamber to create a phase change. The frozen medication becomes a powder. So from liquid to frozen to the powder, making it easier to store and ship. So it also increases the shelf life. So this lyophilization technique is used.
1 minute, let me, can you go back? Let me explain in little detail lyophilization. Actually, lyophilization process is used for the products which are majorly unstable due to the temperature. So the process is what? Up to the formulation, where the API is mixed with the buffer and the excipient is done, and the 0.2 micron filtration is done, and the product is filled in the vials. Now, after filling into the vials, the stoppering is done, but in lyophilization process, the stoppering is not done fully. It is half completed. That means a little space is open between the vial rim and the stopper. Now all this incomplete capped rubber stoppered vials go into the tray into a lyophilizer. So in lyophilizer, there are two functions. First function is the very low temperature, like minus 80 degree, minus 90 degree temperature. So what is done? There is a ramping of lowering of temperature. Gradually, the temperature of these trays where the half rubber stoppered filled vials are kept, and they freeze. Once they are frozen, then there is a application of vacuum. So what happens? Low temperature, where the material is solid and vacuum is very high, the water content of the vial, it starts evaporating. This is called as a sublimation process also. And when entire quantity of water is evaporated, the vials are again fully rubber stoppered and then capped. So this is the process of lyophilization, where the liquid formulated material is filled into the vials, half capped, half stoppered, then frozen to very low temperature, and with the effect of low pressure, the water content of the material is evaporated and the content inside the vial becomes a powder form, which can withstand the temperature during the storage and the usage. So this is what the lyophilization process. Now you can move, Kimna.
But doctor, small question.
Yes.
Rather than vials, means do the liquid formulated liquid is poured into a trays also? Is there any?
No. Trays are used to carry the n number of vials filled with the material.
Bulk trays is mainly to carry the vials only.
Trays are mainly to carry the vials inside the lyophilizer. And there are few questions on the chat box also. Let us. One question is, what is importance of preservative in vaccine? Yes, very good question. Preservatives, what is the role of preservatives? To protect from the contamination. Majorly, the vaccines are not single dose product. They are generally prepared for multi-dose application. So in multi-dose application, what happens, like our COVID-19, everybody must have an experience. The one vial is containing is for the 20 patients, 20 people. So that a doctor or nurse, every time they are piercing that rubber bump to take out the 1 ml or 0.5 ml. So for multiple piercing, there is a chance that the vial get contaminated from any contaminant. So to prevent that contamination, the preservative is very important in vaccines, or you can say where the dosage form is manufactured for multi-doses. Then next is Rakesh has a freeze drying is also used in biosimilar. Definitely, wherever the product is thermolabile, the freeze drying process, freeze drying is lyophilization process. Does that mean lyophilization is extended step in filling for as an in case of injector? Yes, lyophilization is an extended process to make a powder product of the product which are thermolabile. So these are the questions. Now you can continue, Kevna.
So the last step of filling is the stoppering, capping, labeling. The primary packaging is a glass vial. The shells are used with the ram to depress stoppers into the vials. So the primary packaging materials are vials and the stoppers. The vials are stoppered and then it should be crimped onto the system to make the integral container closure. So on the rubber stopper, the crimping is done with the aluminum seal. Finally, the process of crimping an aluminum seal onto a pre-stopper vial is done. So there is a correlation between seal quality and container closure integrity. So after completion of the primary packaging process, the product and container inspection is done, which is a critical part of the filling process. Inspection can be manual, semi-automatic, or the fully automatic. The visual inspection of the vial is done after packing into the primary packaging material. Inspection process is typically used to identify any non-conformities into the batch, such as the cosmetic defects or container closure defects or particulate matter into the container.
Kevna, can you go to the previous slide? I have something to explain. Next, last one.
This one.
Here I wanted to explain this is a very good example how the regulations are getting stringent based on the experience. Earlier days when we were doing filling, so filling operation is always carried out under class A, that is the laminar flow, class 100, surrounded by class B, that is 1000. So earlier days we used to do filling of material into vials and then capping till the vial is open and cap is not fitted completely. We do all operations till in the class A or class 100. And earlier we used to do this cap crimping, the aluminum cap crimping in class C area. But it has been found with the trend that many times after filling, the vials are getting contaminated, though we have capped with the rubber bump. So it has been the observation from the industry and regulators also. And then it is decided that capping, that is the crimping of aluminum seal, should be considered under the class A. So every filling line has been converted into carry out this crimping of aluminum seal inside the class B. So what happened? Primary packing material, earlier we used to call glass foils and rubber bunks as a primary packing material, and aluminum cap we used to call as a secondary packing material. But now, since the cap is also included as a container closure integrity system, the cap is also considered as a primary packing material. That is what the evolution happened in the industry and regulators in the fill finish manufacturing. Now you can continue. There is one question. Let me answer that question also. Powder products finally comes with DS water to use for injecting. What is this DS water, Ankit?
Yes, sir, distilled water.
No, 1 minute. Let me clarify. Distilled water is not used for injectables. Distilled water is not used for injectable. Even I will say distilled water is not at all used anywhere in the pharmaceutical and biopharmaceutical industry except for cleaning of floors.
Sir, deionized water.
It is not deionized water also. Let me
Sterile water for injection.
No, let me explain this. Let me explain this. How many types of waters you are having in your home? Ankit.
Yes, sir.
How many types of water you have in your home?
Sir, normally is the tap water and one is the RO. What we are
yes.
using. Yes.
Now, tap water is used for the cleaning purpose.
Yes, sir.
And for cooking of your food and drinking of water, it is you use the RO water.
Yes, sir.
And that RO water is so important that if you are going to office, you are carrying your bottle. If you are going to a mall, you are carrying that bottle. If you are going with your friend or girlfriend or boyfriend, you are carrying that water.
Yes, sir.
Now see a situation, you have got some fever and you have gone to your doctor. And doctor is asking ki I have to give you an injection. Will you offer that water bottle to doctor? Doctor, can you use this water to give me injection?
No, sir.
Why? You are using that water for cooking, drinking, and 24/7 you are keeping that bottle with you. Why you are not giving that water to doctor to give injection?
Because, sir, there are so many minerals and other things are also in the RO.
My dear friend, you have told me that is RO water. Reverse osmosis water does not contain any minerals and anything. Now let me explain. In pharmaceutical industry, there are 3-4 types of water. One is your raw water coming from your municipal corporation or groundwater. That water is RO passed to prepare a demineralized or DM water. That DM water is used for the cleaning purpose. That DM water is again purified, which makes give you a purified water, which is having a certain specification for microorganisms and pyrogens. That purified water is used for the manufacturing of upstream.
Yes.
Purified water is used up to upstream only. The moment you enter into downstream, where you are gradually purifying your product, you start using water for injection, where the more stringent limits of microbes and pyrogens are there. And when it comes to giving you an injection, it is called sterile water for injection. How this is prepared? The WFI manufactured in a biopharma industry is filled in the vials or the ampoules, and these vials and ampoules which are filled and sealed with WFI are thermo-sterilized in an autoclave.
Okay.
So when you get a injection, powder injection, which needs to be dissolved in a liquid form, you always use sterile water for injection.
Yes.
Next question is, on what stage BT testing done on vials? You see, BT, bacterial endotoxin, which is called pyrogen also. So it starts because in pharma and biopharma industry, water is used, or water is considered as a major raw material. So you start testing your BT from your purified water, WFI, and your drug substance. And when you complete your DP manufacturing, drug product manufacturing, you do final BT testing, plus you have to do BT testing of your vials, rubber bungs, and aluminum caps. So these all are stages where you have to set the BT limits and test that it should be less than 2 lambda limits. Now, what is Ankit Saxena's question? WFI is used. I think I have answered this WFI. Ankit, your second question is TOC level has to be checked. Yes, what is TOC? Total organic carbon. Now, total organic carbon is a specification for your water system. Purified water and WFI. So these are the TOC. Less than 500 ppb is the specification for your purified water and WFI. That your purified water and WFI should not contain total organic carbon more than 500 ppb. Why this specification is put on there? Because at this low level, any chemical testing is not possible. And if your water is contaminated with any of the bacteria which is made up of carbon, if you do TOC testing of your water and you find that TOC levels are very high, you will be able to identify that your water is contaminated. Then you will check the colony count of your water and you will ensure that yes, my water is contaminated. So TOC level is checked on your water system to ensure that the total organic carbon level due to contamination of microbes or any other contaminant should not be high. And the last question, in which application isolator system is used? This is a very good question. Gradually, the industry and regulators has evolved in this isolator technology. Initially, the isolator technology is used, basically isolator technology is used to protect the operator from the occupational contamination. Majorly where the cytotoxic drugs or oncology drugs are handled or manufactured, the all operations were carried out under the isolators. Now gradually what happened, and currently also isolator technology is used in the highly potent hormonal products or cytotoxic products or oncology products. All operations are carried out in isolator. But what happened due to this sterility assurance program of the FDA, LAF or the biosafety cabinets which were used for carrying out the sterility testing, many times it has been found that they are not that efficient to control the sterility. So gradually the manufacturing unit where many batches has been contaminated in the biosafety cabinet or LAF, they have replaced their LAF and BSCs with the isolator technology where there is no chance of going outside air into the inside of BSC or LAF. So this is an application which has been evolved with time, where the isolator has been now used for carrying out the sterility testing. Otherwise, isolator system is basically designed to protect the operators from the occupational hazards. Now you can continue, Kevna.
So next to capping and crimping is labeling of vial. After inspection, labeling of vial. So drug label should having the label which through which we can identify the drug contents, and to state specific instructions or warnings for the administration of the injection, the storage and its disposable. This kind of instruction should be available on the label. As each product having different strength or concentration, same API, but the strength or concentration is different, it must be separately labeled and identified. Labels can be applied directly to the primary container, included as a packaging insert, or applied as a secondary packaging material such as bulk pack box. So final packaging or tertiary packaging. Final, once the product is filled, finished, inspected and packed into the secondary packaging, it needs to be packed into the tertiary packaging for the shipping. The tertiary packaging is used for the protection and the shipping of the product. And packaging must be having label, identify and protect the container for the shipment. Each package is specially designed for the container within. So for example, if different type of packaging form include the vial, syringe or ampoule, so the tertiary packaging is designed accordingly. Cold chain storage and distribution of the product. After packaging into the tertiary packaging, the product is now ready for the transportation. So in most of the biologic therapeutics, they are the temperature sensitive, so they require the cold storage condition during its transportation also. So this is the cold chain storage distribution to ensure that the quality of the precious drug substance and drug products are temperature control supply chain using thermal and refrigerated packaging and storing method is required. So this is the final transportation of the biologics product in the temperature controlled condition or supply chain.
So in the preparation of, we have very well seen the efforts made by the Government of India to ensure the uninterrupted supply chain under temperature control product of this vaccine. I think everybody knows that the kind of efforts has been made by Government of India for the uninterrupted supply of these vaccines. Lot of cold storages and all cold points has been created, cold vans has been procured. So most of the products, they are stored at 2 to 8 degrees centigrade, and they have to be stored and transported at 2 to 8 degrees centigrade. And this is one of the reasons that Moderna and Pfizer's vaccines has not come in India, because their storage temperature is minus 80 degrees centigrade, 86 degrees centigrade. That kind of cold storage supply chain is not available in India. That is why these products has not came to the Indian market, even though the Government of India has given the emergency use approval. And the same thing is happening with the Zydus vaccine and this mRNA vaccine of Pune-based company. because they all also need a storage of minus 70 degrees. So there is one question. Which is the best certification for quality? Which is the best certification of quality of biopharma product, ISO or GMP, which makes manufacturer as world-class facility? You see, in pharmaceutical industry or biopharmaceutical industry, ISO certification has no meaning. You need to comply with, this is a regulatory requirement that you need to comply with good manufacturing practices and acquire the certification, not certification, acquire the approval from the respective regulatory agency based on their standards of good manufacturing practices. For example, one facility is GMP approved by Indian regulator, but cannot qualify for the WHO Geneva. The same thing is happening with Bharat Biotech, that Indian regulators has given the emergency use approval for this Covaxin, but WHO Geneva is still reviewing his documents and the compliances for GMP. Is the extractable leachable studies is mandatory during packaging processes? Yes. Extractable leachable studies are mandatorily required, not for the final product, even for the very early stage of development also. That is why this is one of the major challenge in the use of single-use technology, because every manufacturer, those who wanted to shift to single-use, they have to carry out extractable and leachable studies at each and every step of usage. And they are still very complicated and expensive. Dry ice can be used to store at minus 80 degrees Celsius. Why dry ice is banned in some places? This is from Rakesh Tiwari. Dry ice is used for storage at minus temperatures. And this dry ice is banned because of the presence of carbon dioxide in this dry ice, which is causing a lot of damage to the ozone layer. That is why this carbon dioxide, it is a pollutant, is banned in some of the places. That is why liquid nitrogen is used for the storage. Dry ice generally used for the transfer, short-term usage of the material. Any other questions? Any other questions from the audience? Because I think I have answered all the questions in the chat box. Test already, these questions, Kevna, we have taken this test,
Okay.
so you can skip this test.
Okay. So it's the end of this module.
Should we break for lunch, doctor, or what do you say?
Yes, we can break for lunch. It is 13:32. We can assemble here at 14:30.
Perfect.
We will be here by 14:30.
Yeah, sure.
Okay.
Right, sir. Sure.
Okay.
Bye.
Thank you.
Good afternoon.
Yes, we are here.
Good afternoon, Tirupati here.
Yes, Nilesh.
here.
Yeah, I think let's go ahead and start.
Okay, so my slides are slide is visible now.
Yes.
Okay, okay.
Yes, yes.
So this is the third module where the molecular development, which is the backbone of the biopharmaceutical industry. So we have seen the overview of the molecular development in the previous session, but here we will go in detail exactly how this cloning and all these things are done and how the structure is identified. So we know that the all proteins or the peptides, they are made up of amino acid. So any manufacturer who starts developing a new product, which is a biosimilar, he has to know the structure of the protein, and the structure of the protein is made up of how the amino acids are associated in a series to make a peptide, and then from peptide to polypeptide, and then the polypeptide to proteins. So the most important first part is that the manufacturers, what they do, they suppose I have decided to develop a process for filgrastim or erythropoietin. So I will get this product from the market because that is the only thing available for the development of this new product. So I will get the innovator's product from the market, and first thing I wanted to understand what is the protein, how it is configured. So we will see amino acid sequencing. There are two types of sequencing, N-terminal and C-terminal sequencing. So we will have introduction, what is protein sequencing, history, then determination of amino acid composition, hydrolysis, separation, quantitative analysis, mechanism of protein sequencing, how the sequencing is done. There are two methods, Senger's method, Edmonds method, limitation of Edmonds reaction, then application of protein sequencing. Because from this, once I have the protein sequence with me, either I can design the gene sequence corresponding to that protein, and then which are the institute where the protein sequence can be done, and the conclusions and references. So as we know, the amino acids of proteins, all proteins are formed of 20 amino acids. The naturally occurring total number of amino acids are 20 only. They are mainly formed of alpha amino acids except proline. So I will let you know in next slide what is alpha. They have two functional groups. One is COOH, this is carboxylic group, and this is NH3 is the amino group on the same carbon atom. In physiological conditions, both the groups are completely ionized. This also I will show in next slide. And so an amino acid can act both as an acid and base, which is called amphoteric. So here is that the same amino acid, I have here the structure, the COOH is there and NH3 is there, and both are charged. So CO is minus charged and NH3 is plus charged. So this is called alpha, means on the carbon atom where the functional groups are attached. The first carbon atom where the functional groups are attached. So this C is your alpha carbon atom, and this R, and let me take some pointer. So can you see my pointer now? This R is going to change the amino acids. That is the only thing we will find in all 20 amino acids. So molecules that bear charged group R of opposite polarity are known as zwitter ion. So this structure is known as zwitter ion. It has been established using spectroscopic and X-ray crystal structures. They have properties of ionic compounds. What is the ionic compound property? They have very high melting points. Amino acids are more stable in polar compounds like water. They are very stable in water, and they are in L-isomeric form. We will not go in details of the chemistry of isomers, but there are D and L kind of isomers. So naturally occurring compounds, they all are stable in L forms. So these amino acids are also found in L forms. One minute, I don't know what happened. Okay. Okay. So amino acid sequencing, these amino acids are named based on two conventions. First and the most prominent and privileged convention is three-letter code is transparent. The name has first three letters of amino acid. And the other convention is one-letter code was devised by Margaret Dehoff. Except glycine, all amino acids has a chiral center. Chiral center means asymmetric carbon atom. So these are the 20 amino acids: alanine, arginine, asparagine, aspartic acid, cysteine, glutamic acid, glutamine, glycine, histidine, isoleucine, leucine, lysine, methionine, phenylalanine, proline, serine, threonine, tryptophan, tyrosine, and valine. So the most useful convention of naming of this amino acid is three-letter. You can see these three letters are the first three letter of each amino acid, and this is the corresponding one-letter abbreviation. So we will find this terminology in our subsequent slides. So based on the R groups present, amino acids can be classified as under. There are 5 kind of amino acids. Non-polar aliphatic R group, we have seen. Let me go. This, you have to consider this R group, which is attached to C. So changing this R group, different properties and different amino acids will change. We will see if the R group is non-polar aliphatic R group. Aliphatic means state change carbon compounds. Polar uncharged R group. Aromatic R group. Aromatic R group means there is a cyclic compound like benzene. And positively charged R group and negatively charged R group. Now let us see this non-polar aliphatic R group. What are the amino acids? This glycine, you can see it now here in the structure. 1 minute. Okay. So this now you can see my pointer. So this R group is replaced by H in this CH3, in this CH2, 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 in this CH, CH3, CH3. So these are the aliphatic. These are the straight chain organic compounds. So these all are 1, 2, 3, 4, 5, 6, 7. They all are non-polar aliphatic R groups. Then polar uncharged R group. These are CH2OH, methyl alcohol, and then similarly threonine, cysteine, and this proline, asparagine, and glutamine. You can see the difference in R group is changing, and all these are not polar uncharged. There is no charge on these R groups. So these are another group of amino acids. Then aromatic R groups. Aromatic, I told you there is a ring structure. So this is the benzene ring. This is the benzyl alcohol, and then other tryptophan. Then positively charged R group, you see one of the charged group is attached in the R chain. So there are 3 different positively charged R group amino acids. Then negatively charged R group, you can see COOH acidic group is attached. So there are 2 negatively charged amino acids. So these are the 20 amino acids of various configuration. Now sequencing, how to do the sequencing of protein? Protein sequencing is a technique to determine the amino acid sequence of protein. Basic structure is of protein is like at least 10 amino acids are combined together in a chain. They will form a peptide, and these 10 amino acids peptide, if they get joined together, they will call polypeptide, and then multiple polypeptide, if they will combine, they will make a protein. So it is a method to understand the structure and function of proteins in living organism. The protein sequencing is to understand the structure and function of the protein in living organisms. Amino acid sequence determines the eventual 3-dimensional structure of the proteins. The proteins, if they are not 3-dimensionally configured, they are not called active proteins. So to have an active protein, this 3-dimensional structure should be known to us, and that can be done by the amino acid sequencing. So I have already told all proteins are polymers of amino acid, and all these amino acid except to have one amino group attached to the carboxyl group. What is protein sequencing? Protein sequencing is the technique to determine the amino acid sequence of a protein. Who are the main contributor in this sequencing of protein? One is Edman. In 1947, what he has done, he found the method to decode the amino acid sequence of a protein using chemicals. And second is Sanger. In 1955, he has got the Nobel Prize. He was able to present the complete sequence of insulin. So you can understand that this sequencing of protein is not a new thing, and insulin's structure was known in 1955. Determination of amino acid composition, what is involved? Amino acid composition and purity must be known before starting sequencing. That means the protein which we wanted to know the structure, it should be highly pure compound. And what is the amino acid composition? That means what is the number of moles of each amino acid present in the protein. The polypeptide chains of multimeric proteins should be separated, and molecular weight of each chain should be measured. Polypeptide chains of multimeric means, as I have told you, peptides are made up of amino acids and polypeptides are made up of peptide chains. When many polypeptides come together to form a protein, that is called multimeric polypeptide. So what we have to do, these multimeric polypeptide chains, they should be separated first, and they have to be straightened first before going for the amino acid composition. The determination of amino acids is done by hydrolysis. Hydrolysis means cutting from a chain. Then the separation means the portion which has been cut out from the chain is separated, and then the separated part is quantified. That is, these 3 steps are involved in the determination of amino acids. Now sequencing, protein sequencing, how we do? Technique to find out the sequence of amino acid in protein. There are 2 kinds of sequencing. Now let me go to the previous slide once again, one more time, so that it is very clear to you. What we have seen in this amino acid, now you imagine one after one amino acids are joined together to make a peptide, and then peptides are making polypeptides. So what will happen when we straighten the polypeptide? One side we will get this NH3. So this is called N terminal. And the other part we will get COOH. So this part called C terminal of a polypeptide or protein chain. So the sequencing is possible from both the ends. So if we are doing sequencing from N terminal, NH3 group, then it is called N-terminal sequencing, which is done by Edman degradation. And if we will do the sequencing from C-terminal, COOH group, it is called C-terminal sequencing. And what is the advantage? As I told you, I have decided to make a biosimilar of filgrastim. So if I know the amino acid sequence of filgrastim, from both the ends, N and C terminal, I will be able to predict the DNA sequence. And if I know the DNA sequence, I will reconstruct that DNA sequence. Then I will be able to make my clone. So this is the backbone of the biopharmaceutical industry, knowing the sequence of protein, prediction of DNA, construction of DNA, and then cloning. We will come to the cloning. So let me see, very briefly, we will see the Edman degradation, that is N-terminal sequencing. What are the steps involved? Purification of protein. But as I told you, what will happen if the protein is not pure? The amino acid which we are cutting and identifying and quantifying, if it is contaminated with any other protein, the amino acids of that contaminated protein will come in my structure. So I will not be able to reconstruct the right DNA. So that is why the 100% pure protein is required. Then protein has to be denatured, which we will see. I told you that it has to be straightened. Because once we will straighten it out, we will cut from one end, one by one amino acids. Then N-terminal, the protein digestion, then N-terminal labeling, then separation of labeled amino acid by chromatography, then detection through mass spectroscopy, and then the data analysis. So now what is involved in purification? We all know that in the lab, very common technique is the SDS page. Sodium dodecyl sulfate polyacrylamide gel. We run the protein sample on this SDS page, and based on the molecular weight, the proteins are separated on the gel, on the plate. So this can be done on the two-dimensional gel. First you run like this, and then whatever bands are separated, you run like this. So you will get the most purified protein of interest on your gel. From this gel, you have to take out the protein. This is your protein. Yeah, this is protein before SDS, sodium dodecyl sulfate. So this is the coiled protein. Now what we have to do, we have to straighten this out. Both the terminals has to be straightened. So SDS role is, after SDS, it is straightened out. This is called denaturation of the protein. So protein of interest is immobilized by being absorbed onto a chemically modified glass or an electro blotting onto a porous PVDF membrane. So this is the two-gel SDS page chromatography where you can do the purification. So you have got purified, denatured, straight chain of your protein. Now next stage is hydrolysis, denaturation. By heating a sample of protein in 6 molar HCl up to 100 to 110 degrees Celsius for 48 hours, it may degrade some amino acid. So what will happen? Because we need to add 6 molar HCl during our N-terminal sequencing. So to avoid this thing, we need to add certain reagents like thiol reagents or phenols are used, and then performic acid, HF for intrachain or interchain SS bond, because they are joined with SS bonds that has to be broken before straightening. Then protein digestion. Now protein chain is available with the amino acid. Now use endoproteinases lysine C, CNBR, pepsin or trypsin to digest proteins into a population of peptides. So the complex structure of protein is broken down into peptides, small peptides. Other enzymes include gluC and chymotrypsin. Add enzyme at a ratio of 1:20, enzyme is to protein ratio. Incubate at room temperature for 6 to 9 hours. For better results, use mixture of these enzymes to get more number of small peptides. Now what we have to do? N-terminal labeling. Labeling means we will add something on the N-terminal of first amino acid of the peptide. In the admin reagent, phenyl isothiocyanate, PTC, is added to the absorbed peptide together with a mildly basic buffer solution of 12% trimethylamine. This reacts with the amino group of N-terminal. The terminal amino acid can then be selectively detached. Then what we will do that this PTC attached amino acid will be cut, and then derivative, then isomers to give a substituted phenylthiohydrantoin, which can be washed off and identified by chromatography, and the cycle can be repeated. Now let us see. You see, this is our amino acid sequence, where this is one amino acid, this is second amino acid, this is third amino acid. And this is what? Admin reagent, phenylthiocyanate. So what will happen when we will do mixed alkaline condition pH 9 with these two reagents? This admin reagent will attach with this N. So this is formed. Now the structure is like this. Now what we have to do? We have to cut it from here. So for cutting anhydrous HF, if we will use, what will happen? This will be cut from here. So this NH group is with the remaining of the peptide. And this first amino acid has come with this admin reagent. Now this admin reagent can be isolated through chromatography and can be quantitated by HPLC method. So this is the N-terminal sequencing. What will happen after this? After this separation, this PTH or the admin reagent will again come here and will attach with this NH group, and then it will cut from here, and then this second amino acid will be released. So by this one by one amino acids will be released in the form of PTH amino acid phenylthiohydrantoin. And this compound individually, they can be collected on the RP-HPLC, and then they can be quantified by the HPLC and the LCMS. Now, what is C-terminal? The same process, what we have to do, we have to start from here. Instead of starting from N-terminal, we have to start from C-terminal. So C-terminal definition, this C-terminus also known as carboxyl terminus, which I have already explained. Carboxyl terminus, C-terminal tail, C-terminal end, or COOH terminus. This is the end of an amino acid chain, protein or polypeptide terminated by a free carboxyl group COOH. You see, this is more clear. This is N-terminal. This is a polypeptide or a peptide. where this is one amino acid, this is second amino acid, this is third amino acid. So if we will start analyzing the amino acid sequence from here, because here this is up to this, there is a one amino acid. This is NH2 group and this is carboxyl group. So we have to analyze this. If we will start from here, this will be N-terminal sequencing. If we will start from here, this will be called C-terminal sequencing. So by this entire protein, one by one, each amino acid is structurally, quantitatively estimated, and then we can make a sequence of amino acid like this. You see, alanine, threonine, glycine, and then we will know the structure of peptides. And if we will combine again the structure of peptides, we will know the structure of polypeptide. And if we combine all polypeptide, we will know the structure of protein. So this is what called amino acid sequencing, the first step in identifying the sequence of a protein. Now, the peptide is hydrolyzed into its constituent amino acid by heating into 6 molar HCl, and then the separation by chromatography or dialysis. And then the quantitative analysis of the amino acid residues with Edman reagent of peptide reacts quantitatively with ninhydrin. Ninhydrin, one of the reagent which attaches with this peptide amino acid residue. On heating, an amino acids react with 2 molecules of ninhydrin to yield an intensely colored product. The purple, most of the amino acids, when they react with ninhydrin, they give purple color, except proline gives yellow color. So this is what the amino acid sequencing. Now, mechanism of protein sequencing, we have already discussed Senger's method and Edmonds reaction. Senger's method is what? Frederick Sanger was a Nobel laureate. He showed for the first time that amino acids are covalently together with alpha amino and alpha carboxyl groups. He suggested a stepwise release and identification of amino acid starting from N-terminal. So this Sanger has started and got the Nobel Prize for the sequencing of protein. He used a reagent fluorodinitrobenzene, FDB, which is commonly called Sanger's reagent. The FDB reacts with free amino group of N-terminus. Upon hydrolysis, a yellow-colored dinitrophenol DNP derivative of N-terminal amino acid is produced. The DNP amino acid is identified, comparing it with a known standard of DNP amino acid. That means that DNP amino acid standards are available commonly in the market. So when we are doing N-terminal sequencing, we use standards of DNP amino acids to quantify and identify each and every amino acid which are cutting and getting separated, The densyl chloride, another reagent, is commonly used because it forms an intensively colored derivatives that can be detected with high sensitivity that the dinitrophenyl compound. It reacts with the unchanged, you see difference between this Sanger's reagent FDB and densyl chloride. Densyl chloride can react with C-terminal also and N-terminal also. So this can be used for sequencing from C-terminal to form a sulfonamide derivative that is the stable under condition that the hydrolyzer peptide bond. Although the densyl method for determining the amino acid terminal residue is sensitive and powerful, what are the disadvantages? It cannot be used repeatedly on the same peptide because the peptide is totally degraded in the acid hydrolysis step. Now, second is Edman degradation. In 1950, Edman developed a method of protein sequencing. It involves sequential identification of amino acid from N to C terminal. This is mainly from N to C terminal. That phenyl isothiocyanate reagent is used for the Edman degradation. The amino terminal N-terminal residue of the protein can be identified by reaction the protein with the PTC that forms a stable covalent link with the free amino group prior to hydrolysis with 6 molar HCl. The 6 molar HCl is used for the hydrolysis once this PTC is attached with N-terminal of amino group. Phenyl isothiocyanate reacts with the unchanged N-terminal amino group of the peptide to form a phenyl thiocarbonyl derivative. The labeled N-terminal amino acid can be identified by comparison of its chromatographic properties with standard fluorodinitrobenzene and densyl chloride. Then under mild acidic, the cyclic PTH phenyl thiohydration of the terminal amino acid is liberated, which leaves an intact peptide shorted by one amino acid because it has taken out only the N-terminal one amino acid. The released PTH amino acid is identified by high performance HPLC chromatography. The sequencing technique has been automated. Now automated sequencers are available and refined so that upward of 50 residues from the N-terminus of a protein can be sequenced from picomolar quantities of material. Now, you see, this is the diagram. You see, this is the 5 amino acid. So in first round of reaction, what happened? This first amino acid is attached with Edman reagent. This is phenyl isothiocyanate. This is the Edman reagent. This is your alanine, first amino acid. This is attached here with this first amino acid, and then hydrolysis. This is cut from here, and this is PTH alanine. This is first amino acid is released. Now in second round, what will happen? The second one will come to the terminal. Then second, this phenyl isothiocyanate will react, and this will release the glycine. So same way, this kind of individual amino acid derivatives will be released from your peptide, and they will be quantified by HPLC method, and then you can make a sequence of your protein key in which sequence these amino acids are attached together. Limitations of Edman reaction: the Edman degradation proceeds from the N-terminus of the protein. It will not work. The N-terminal amino acid has been chemically modified. If this N-terminal is modified by any means, this reagent will not work. It also required the use of either guesswork or a separate procedure to determine the position of disulfide bond. What is disulfide bond? In some of the amino acid, we have seen the sulfur group. So whenever there are two sulfur groups together, they will make a double bond between two sulfur groups. So this two sulfur groups is called disulfide bond, which is not broken by this reaction. Application of protein sequencing, where it is applicable: knowledge of the sequence of amino acids in a protein can offer insights into its three-dimensional structure and its function in cellular location. So this is one of the application. Certain amino acid sequences serve as signals that determine the cellular location, chemical modification on and half-life of a protein. This is why we have seen during our this COVID-19 journey that whenever there is a variant reported, I think everybody has seen that this is in news. Whenever there is a variant reported, immediately Government of India has started the complete sequencing of that variant. So this variant sequencing, if you know the location of amino acid has been changed, you will know what kind of variation has happened and what are the functions that variation will going to produce. Protein sequence can elicit the history of life on Earth. That means how the virus is modifying, this can be determined from the amino acid sequencing also. Which are the institutes? In India, Institute of Genomics and Integrative Biology, IGIB, New Delhi, and the Center for Genomic Application, TCGA, Kolkata. They are doing this sequencing. Outside India, Novo Nordisk Foundation Center for Protein Research, University of Copenhagen, Institute of Protein Research of the RAS, IPR RAS, Moscow is doing the protein sequencing. Nowadays, a lot of protein sequencing, even some of the biopharmaceutical industries, they have installed all these facilities to do protein sequencing in-house. Now, protein sequencing is a technique to determine the amino acid sequence of a protein. Conclusions, what are the conclusions? It also gives information regarding which conformation of protein adopts three-dimensional structure. Discovering the structures and functions of protein in living organisms is an important tool for understanding cellular processes. It allows drugs that target specific metabolic pathways to be invented more easily. The first protein sequencing was achieved by Frederick Sanger in 1953. He determined the amino acid sequence of bovine insulin. Sanger was awarded the Nobel Prize in 1958. Now, we know the sequence of our protein. Now we wanted to construct a gene. But if we are able to construct a gene, we have another exercise to establish that whatever sequence I have constructed is correct sequence. So there is a second requirement in the industry, that is the gene sequencing. Any question in this protein sequencing, amino acid sequencing? I think somebody is having a question. These PTCs are not fluorescent. They are producing a visible range color. So I think there is no question. So let me proceed with the gene sequencing. So first we should see what is DNA. Briefing on DNA. DNA is found in the cells of all living things. So this is, you can consider our cells. Inside the cell, we have a nucleus. And inside the nucleus, we have a chromosome. And in chromosome, the genes are situated. So if we come from the cell to nucleus to this chromosome, and this chromosome is coming like this nucleosomes, and nucleosomes are converted into DNA double helix, which I think you will find in every biopharma industry's logo. And these double helix DNAs are made up of, we call this base pairs. So DNA contains all the genetic information that makes you who you are, and every individual organism has unique DNA like a fingerprint. It contained phosphorus in the, now we are talking about this base pair, because that is the building block of DNA. It contains phosphorus in the form of phosphate. So first component of building block of gene is phosphate. Second is deoxyribose nucleic acid. These are the sugars. You will find two kinds of sugars. ribose sugars and deoxyribose sugars. It is a molecule that encodes the genetic instructions. Most DNA molecules are double-stranded helix. Each molecule consists of 2 long biopolymers. We will see all structures. So I have a lot of diagrams to explain these things. Made up of simple units called nucleotides. These base pairs are called nucleotides. Each nucleotide is composed of a nucleobase and sugar and phosphate. Nucleotide is a composition of nucleobase, sugar, and phosphate. And nucleobases are known as, right now you just remember GATC. DNA is well suited for biological information storage. DNA stands for deoxyribonucleic acid. Term DNA was given by Zachirs. DNA is biopolymer, consists of nucleotide as monomeric unit. So nucleotide is the base which we are talking about. DNA is a double helical structure in eukaryotic cell like animal cells and prokaryotic cells like bacterial cells. But in viruses, it may be a double-stranded or single-stranded. We will see single-stranded also. And presented as monopartite or multipartite. So the DNA vaccine is prepared using single-stranded DNA, which is equivalent to mRNA. We will see in coming slides. In eukaryotes, DNA is presented in nucleus surrounded by nuclear membrane. In prokaryote, there it is a difference between animal cells, bacterial cells, and virus. Eukaryote DNA is present in nucleus surrounded by nuclear membrane. You see, this is nuclear membrane. Prokaryotic DNA is present in nucleoid region of cytoplasm. Generally, in bacterial cell, this DNA is present in this cytoplasm, which are called plasmids also. Without nuclear membrane. In prokaryotic, we have this nuclear membrane. In eukaryotic, we have a nuclear membrane. In prokaryotic, we do not have this membrane. These all DNAs are in cytoplasm. And in virus, DNA is presented in the core of virus. Only virus DNA will be there. One DNA will be there. And a protein layer. A protein layer called capsid. Very simple structure. Now, what is the DNA? Is a deoxyribonucleic acid is the hereditary material in humans and almost all other organisms. Nearly every cell in the person's body has the same DNA. So, every cell is having the same constitution of DNA. Where it is located, most DNA is located in the cell nucleus, where it is called nuclear DNA. But a small amount of DNA can also be found in mitochondria, where it is called mitochondrial DNA. Now, what is the structure of DNA? The structure of DNA is illustrated by a right-handed double helix with about 10 nucleotides pairs per helical turn. I will explain this thing. Each spiral strand composed of a sugar, I have told you, phosphate backbone and attached bases, is connected to a complementary strand by hydrogen bonding covalent between paired bases adenine A, thymine T, guanine G, cytosine C. Now let us see this structure. This is the structure of DNA. As I told you, there is a sugar, deoxyribose sugar, a pentose sugar. Why pentose? Because there are 1, 2, 3, 4, 5 carbon atoms. This is the sugar. This is the phosphate. And this is the base, ATCG, adenine, thymine, cytosine, and adenine, thymine, cytosine, and guanine. These are the 4 different base pairs. So this is the nucleotide, the building block of DNA. Now let us see the structures. This is the pentose sugar. Now we are calling DNA and RNA. DNA means deoxyribose sugar. RNA means ribose sugar. Let us see what is the difference between DNA and RNA. The sugar, this is ribose sugar. Ribose matlab, if you will see this OH group, in this one oxygen is extra. If I remove this oxygen, this will become 2-deoxyribose sugar. You can see here. Both the sugars are same. In RNA, we get this sugar where the sugar is oxidized. In DNA, the same is deoxidized. This is the basic difference between, one of the basic difference between RNA and DNA. Now let us see the bases. 4 bases we have seen. They are divided into 2 groups. One is pyrimidines, other is purines. Pyrimidines made up of 6-member ring. These 6-member ring, thymine and cytosine, T and C. Purines are made up of 6-member ring fused with a 5-member ring. This is adenine and guanine. These 4 base pair we are talking about DNA. First difference we have seen in RNA and DNA is the difference of oxidation of sugar molecule. Second difference we will see in this bases. There are 4 kind of bases in DNA: thymine, cytosine, adenine, guanine. They are divided into 2 classes: pyrimidines with 6-member ring and purines. 6-member ring is fused with 5-member ring. The rings are not only made up of carbon-specific formulas, and structures are not required for inclusion bodies. Now you see, this is the structure of RNA and DNA. The C is in this 5-member ring. Carbon atom, 5 carbons are there. So we start calculating the number from where the hydroxyl group is attached. You see here. So this is 1-number carbon, 2-number carbon, 3-number carbon, 4-number carbon. Why it is? Because this is the IUPAC nomenclature rule that counting of carbon atoms should start from a free functional group. So this is one of the free functional group from where 1, 2, 3, 4, 5 is calculated. In the same manner, in this also it is calculated. So you see, this is the 5-member group. The C is named 1 to 5, 1, 2, 3, 4, and this is the 5th one. From this 5th one, the phosphate group is attached. So this will be called 5-N. And from this, another phosphate group is 3rd number. So one phosphate is attached on the 5th group, and the other is attached in the 3rd group. So we call it 5 to 3, 5-end and 3-end. You can see here, this is 5-end and this is 3-end. This is 3rd number carbon phosphate group is attached. Now this phosphate group, and on the number 1, what is attached? The base. What is base? ATCG. So this is completely called, you can see below, this is completely called where phosphate, sugar, and base all are present. This is called nucleotide. and without phosphate, it is called nucleoside. So in RNA, what will happen? Here you will get oxidized OH, and in DNA, you can see deoxyribose. Oxygen is removed. Otherwise, the structure is same. The next difference between RNA and DNA we will see in the bases. Okay. Now see what is changed. These are the pyrimidines, cytosine and thymine in DNA. And below is the purines. Pyrimidines are 6-membered rings. Purines are 6-membered ring fused with 5-membered ring. So these are the adenine and guanine. So there is no change in purines, adenine and guanine in RNA and DNA. But there is a change in pyrimidines when it comes for RNA and DNA distinction. The thymine is replaced by uracil. So in DNA, the pyrimidines are cytosine and thymine, C and T, and purines are adenine and guanine. But in RNA, pyrimidines are uracil and cytosines. So wherever we will find T in the DNA, in the corresponding RNA, we will find U in the RNA. This is the basic fundamental difference between RNA and DNA. You can see this basis. This is pyrimidines, cytosine, uracil, thymine, and phosphate group. And this is deoxyribose group. These are the nitrogenous bases. And this is phosphate group. And this is sugar group. Clear? Now let us come to the structure of DNA. We have seen this is double helix. So this is called one arm and this is called second arm. So this is called double helix, double stranded DNA. Now let us see the properties of this DNA. So the sugar phosphate backbone, we have seen that sugar phosphate backbone, this one, this sugar phosphate. This sugar phosphate is on the outer side and base is in the inner sides. See, this is sugar phosphate backbone. And inside you can see A, C and TG. So A is always combining with T and C is always combining with G. You will see this structure also in very detail. It consists of 2 helical polynucleotide chain. This is 2 helical of polynucleotide chain. 2 polynucleotide chain coil around the same axis to form a right-handed double helix. This is right-handed double helix. In the helix, the 2 chain or strands are anti-parallel, that is having an opposite polarity. They are having the opposite polarity. Backbone of each chain, which consists of alternate sugar phosphate residue, hydrophilic. This is phosphate is present, that means it is hydrophilic, are on the outside of the double helix facing the surrounding. The purine and pyrimidine bases of each strand face inward towards each other. You see this one. You can see this. These are the purine and pyrimidine bases. They are facing each other inwards. The bases are stacked perpendicular to the long side axis of the double helix. They are perpendicular to the long side of the double helix. This base pair are 0.34 nanometer apart in DNA helix. This distance. If you see the distance between two base pairs, you see this one. This distance is 0.34 nanometer apart. A complete turn of helix, this complete turn of helix takes 3.4 nanometer. So how many base pair are in one helix? 10 base pair are present. In one helix, there are 10 base pair are present. The external diameter of helix, this external diameter from here to here, this is 2 nanometer. The helix has two external grooves, one groove and the other groove. The narrow groove is called minor groove, this one. What is the importance of this nano groove, minor groove? While the wide groove is called major groove, this groove, what is the importance? The major groove is site for DNA binding protein. Any protein going to bind with DNA will be in this major groove. And the small molecules will bind on these minor grooves. Now the pairs of bases are always between a purine and pyrimidine. That means if this arm is having purines, this arm will be having the pyrimidines. Specifically, the pairs are A will combine with T and G will combine with C. So if A, T, A, G, C, this sequence is coming here because each helix is having 10 pair, 10 nucleosides. So you will find A will combine with T and G will combine with C, which are the base pairs found in char graft. There is hydrogen bonding between the bases. This is the hydrogen bonding. A with T, C with G, G with C, A with T with A, G with C. So between A and T, there is always two hydrogen bonds. Two hydrogen bonds between A and T and three hydrogen bonds between G and C. This is 3, 1, 2, and 3. So this is phosphate, this is sugar, this is bases, and this is one helix, this is second helix, and they combine and they can form double helix DNA. Two chains do not have the same base composition, not identical, but two chains are complementary. These two chains are complementary to each other. A is combined with T, so T will combine with A. C is combined with G, so G will combine with C. So these two chains are complementary chains. Such chains are called as complementary chains. The first chain, for example, AT, AC, GC, AC, then 3A, 1T, 3C, 1G, then second chain will be complementary to each pair, TA, TC, G, G, TG, 1AT, 3T, 1C, 3G. So this is the complementary chains. This is a double helix, this is one helix, this is second helix. A sugar and a phosphate backbone connects nucleotides in a chain. This is a chain. Hydrogen bond between paired bases hold and two DNA strands together. This is the hydrogen bonds. DNA strands are antiparallel. Antiparallel means if one is 5 to 3, the other one is 3 to 5. This 5 and 3 we have seen the attachment of this base to the sugar. In this, it is attached to the fifth. In this, it is attached to the third. This is again another, this is anti-parallel, 5 to 3. This is parallel, 3 to 5. The same chemical structure. This is phosphate, this is sugar. deoxy sugar because OH is there. These are the bases, purines and pyrimidines. And this is hydrogen bonds, 2 hydrogen bonds between A and T and 3 hydrogen bonds between C and G. So this is the structure of DNA. Now what will be the structure of RNA? Here this O will not be there and C will be replaced by U. This is the, now gene sequencing. This is the, now what we have to do for gene sequencing. This is the polymerized structure of DNA. If we will zoom into the DNA double helix, this will look like that. And if we go inside this helix, then it will look like this: the phosphate, the sugar, the base pairs, the hydrogen bonds. So this is the structure of DNA and RNA. And another difference in DNA and RNA is DNAs are found in double helix. RNA is only one sequence, one helix. It is never converted into a double helix. Now, this is the, if we want to do sequencing, now let us go to the gene sequencing. The principle is same as we have done with the amino acid sequencing and terminal sequencing. DNA, we have to break it. So denaturation, this is called DNA denaturation. How it is achieved? The DNA denaturation is the separation of double strand into 2 single strands. This is a double strand. If I will heat it, simple heating will denature the DNA. If I will heat it, it will start the hydrogen bond. These hydrogen bond will start breaking. And these 2 different strands will separate out. If I will heat more, this will be completely denatured. Both these strands are separated with their phosphate, sugar, and nucleotide bases. And if I will again cool it, they will again form the hydrogen bonds as they were having initially. So this is the simple procedure of denaturing of DNA. Now there is one industrial application of this. We have seen that the vaccines are manufactured using live vaccines and the attenuated vaccines, DNA vaccine and mRNA vaccines. So attenuated vaccines or the live vaccines and the messenger RNA vaccines, they are very, very stable. Now you can identify why Pfizer's and Moderna vaccines are required to be stored at -70 degrees. Because DNA is very sensitive to heat. So this RNA or DNA vaccines, especially in DNA vaccine, when this DNA is formed, which is responsible for the antibody of the viral gene, there is a very critical step in the manufacturing process, within how many minutes that the entire solution should reach to 228 degrees centigrade. That is why we are seeing the delay in coming of DNA vaccine in the market. They are not stable at 228 degrees centigrade, first thing. Second thing, because of the sensitivity towards the temperature of this DNA, when this DNA vaccine is manufactured, there is a step where these 2 arms need to be folded is very critical for temperature, and managing that in process control is very, very difficult. That is the reason DNA vaccine first time manufactured in India is taking so long time. I cannot disclose the proprietary information in this webinar, but yes, I know the all details. That is the reason instead of getting the DCGI's approval for this vaccine, the vaccine is not available in the market. Now, how the protein synthesis is done? Because what is happening? Protein synthesis is why it is required. When viral vector or the DNA sequence or the mRNA sequence inserted into our body, antigens are inserted and our body start responding to with the antibodies. What are these antibodies? These antibodies are nothing but the proteins. So protein is manufactured in body or outside body, which is we call monoclonal antibodies also. They are their structure, their sequence, the amino acid sequence information is stored here in DNA. What will be the amino acid sequence? Now see why this is very important in biopharmaceutical to understand. Suppose I wanted to manufacture a protein whose DNA sequence is from here to here. And this gene I have artificially developed. Now, artificially developed this fragment of gene will not produce the protein until unless it is integrated into this DNA. So that is why we need to know the protein sequence, exact protein sequence, and then corresponding gene sequence, then construct that gene sequence and integrate into DNA. Then the process of developing of antibody will start. Whether it is in vivo inside the body or we are doing a manufacturing a biosimilar. What we are doing in biosimilar, that artificially reconstructed gene sequence, we are inserting into a vector, and that vector is getting inserted into that gene, and that gene is having a further process of producing the antibody, that is monoclonal antibody. So we will see how the monoclonal antibodies are produced from this DNA to mRNA to protein. Now, in the protein synthesis, the same process is followed whether it is in vivo or in vitro, or you can call it in biosimilar processes. Once gene is constructed, inserted in the vector, and the vector is inserted in the host cell. Host cell means CHO cell or viro cell or bacterial cell. Then this process will start for the manufacturing of monoclonal antibody, which are protein in nature. So there are transcription, genetic code, transfer RNA, ribosomes, translation, and polyribose. These are the steps involved once we have a genetically modified host cell or cell line, which we are calling RCB or WCB or MCB. Once we are ready with our WCB, this process we do in vitro inside the fermenter to manufacture multiple copy of same protein so that product of interest is available in desired quantity. Next. Anybody is having any doubt or question, please feel free to clarify here itself, because we have seen the amino acid sequencing and importance of amino acid, then gene sequencing. Now we are going to synthesis of protein, which is our main objective, ki how this monoclonal antibody or the biosimilars are manufactured. Anyone is having any questions? If no, then I will move forward. So first is transcription. Transcription or RNA synthesis. Now see, this DNA will not manufacture the protein. Protein manufacturing is done by always RNA, and that is called mRNA, messenger RNA, which is having the codes for individual amino acids. So first thing is that this DNA should prepare a complementary RNA, which will be responsible for the manufacturing of protein. That is why I told you ki what is the difference. mRNA based vaccines, they are readily available. mRNA will go inside our body and start manufacturing the antibodies or the proteins which are neutralizing the viral infection. In DNA vaccines, DNA will go inside the body. It will do this transcription to manufacture RNA, and RNA will then produce the antibodies which are going to neutralize the viral vector. RNA synthesis is the process of creating an equivalent RNA copy of a sequence of DNA. See here. What will happen? This RNA will get open up, and certain enzymes present, they will start creating a complementary to this strand. For example, so suppose here you see, we can see this thing. This DNA is opened up. For example, these 1, 2, 3, 4 are our genetic codes for manufacturing of protein. Now this DNA will open up with the help of enzymes. Then mRNA construction will start. This phosphate, deoxyribose sugar, and complementary nucleotide base. What will come? T. T will be attached. So one nucleotide will be manufactured here after breaking of this. So one nucleotide for this, another nucleotide for this, another nucleotide for this. So this way we will get a single strand mRNA, which is complementary to this one with the difference of oxidation in sugar, and C is replaced by U. And this sequence which is constructed here will be called as a RNA. You will see that it is a equivalent, creating an equivalent RNA copy of a sequence of DNA. This sequence of DNA. Both RNA and DNA are nucleic acids which use base pairs of nucleotides as a complementary language that can be converted back and forth from DNA to RNA. Now see, we have seen that here we have created a complementary copy of RNA here. Here, you see. And that complementary copy will be equal to what? This sequence. A will be T, C will be U. This sequence. So in DNA, this copy is creating the mRNA, and this copy is equal to the mRNA. You will see these things. They can be converted back and forth from DNA to RNA in the presence of the correct enzymes. Now, this process which we have seen right now, DNA transcription, it will transcribe RNA, and RNA will translate the codes available in the sequence, these codes. We will see what are these codes, into protein. This is called central dogma. The basic fundamental of molecular biology and protein synthesis. Any confusion? We will see how the protein, in future slides, you will see the examples of how the proteins are constructed. If no questions, let us move forward. So DNA is transcribing RNA, which is a complementary. With the changes, we know the oxygen deoxyribose and ribose sugar difference, and the pyrimidine difference, C is replaced by uracil, and DNA will be a double stranded, RNA will be a single stranded. These are the three major differences in DNA, RNA, and RNA will be having these codes for amino acids, which will convert into protein, that is called central dogma. Now next is, during transcription, a DNA sequence is read by RNA polymerase. So this is the enzyme. Every activity in this is controlled by the enzyme. So these are the different enzymes we will be seeing. RNA polymerase, matlab that will polymer these nucleotides, which produces a complementary anti-parallel RNA strand. Anti-parallel, we have seen that these two are anti-parallel. A is combined with T and C is combined with G. As opposed to DNA replication, transcription results in an RNA complement that includes uracil in all instances where thymine would have occurred in a DNA complement. Transcription is the first step leading to gene expression. This is what? The expression of gene. The stretch of DNA transcribed into an RNA molecule is called a transcription unit and encodes at least one gene. So in this entire length of DNA, you will find a selective area which is responsible for, which is having the information for a particular protein or antibody. So messenger RNA will be reconstructed in that region only. There is no need to open entire DNA helix. This is controlled by other RNAs, which part of the DNA to be opened with the help of enzymes, and the complementary messenger RNA and the regulatory RNA are transcribed. Transcription unit encodes at least one gene, and this gene is responsible for particular antibody or protein. If the gene transcribed encodes for a protein, the results of the transcription is messenger RNA. which will then be used to create that protein via the process of translation. So from messenger RNA to manufacturing of protein, this procedure is called translation. Why, we will see in the coming slides. Alternatively, the transcribed gene may encode for either ribosomal RNA, we have seen is messenger RNA, which is responsible for translation of protein. Other RNAs which are helping for in this translation are rRNA, ribosomal RNA, and transfer RNA, tRNA. Why these two are important? Because protein synthesis will happen on ribosomes. So to take messenger RNA from this site, the messenger RNA is constructed here. From this side, that messenger RNA has to go to ribosome. So this transfer RNA will have the information so that messenger RNA is transferred to ribosome. And ribosomal RNA will help in translation into proteins. The DNA transcription unit encoding a protein contains not only the sequence that will eventually be directly translated into a protein, that is called coding sequence, but also regulatory sequences which are rRNA and tRNA that direct and regulate the synthesis of that protein. As in DNA replication, DNA is read from 3 to 5 during transcription. We have seen here 3 to 5. From here to here, transcription means complementary chain. Meanwhile, the complementary RNA is created from 5 to 3 because it is in the opposite direction. Only one of the two DNA strands called the template strand. What is template strand? If this blue strand is making mRNA, then this will be called template strand. And if we see here, suppose this is a template strand which is converting into messenger RNA. If we see the structure of messenger RNA, this will be same like this, except the difference of uracil. So this is called template strand, and the other one will be called template strand is used for the transcription. The other DNA strand is called a coding strand. This is a coding strand. This is a coding strand. Sorry, this is a template strand, and this will be called as a coding strand because the structure of this strand will be exactly same as messenger RNA except the difference in sugar and uracil. So these are called, the other DNA strand is called the coding strand because its sequence is the same as the newly created RNA transcript except for the substitution of uracil for thymine. Now, transcription is divided into 3 stages: initiation, elongation, and termination, because we need only that much of fragment of messenger RNA which is coding and regulating. So there should be some site in the entire DNA, like some site in the entire DNA from where that messenger RNA will start then elongate, and then terminate. It is not required that entire DNA has to be converted into messenger RNA. Wherever this coding is done, template is there, that part will be opened up, initiated, elongated, and terminated. So initiation in bacteria, transcription begins with the binding of RNA polymerase to a promoter in DNA. RNA polymerase is the core enzyme consisting of 5 subunits, 2 alpha subunits, 1 beta subunits, 1 beta dash subunit, and 1 omega D subunits. We will not go in detail of this structure. At the start of initiation, the core enzyme is associated with a sigma factor that aids in finding the appropriate 35 and 10 base pairs downstream of promoter sequence. That core enzyme finds the site from where it has to be started and it binds there. Then elongation, one strand of DNA, the template strand for non-coding strand or non-coding strand, is used as a template for RNA synthesis. One by one, nucleotides are getting assembled in sequence. As transcription proceeds, RNA polymerase transfers the template. Transverse means, jaise we will go inside, that polymerase will help the assembly of nucleotides in the messenger RNA. Transfers the template strand and uses base pairing complementary with the DNA template to create a RNA copy. Although RNA polymerase transferase and template strand starts from 3 to 5, the coding non-template strand and newly formed RNA can also be used as reference points. So transcription can be described as 5 to 3. This procedure, this produces an RNA molecule from 5 to 3, an exact copy of the coding strand, the other strand, except that thymines are replaced with uracil, and the nucleotides are composed of a ribose 5-carbon sugar, where DNA has deoxyribose, one less oxygen atom in its sugar phosphate backbone. Unlike DNA replication, messenger RNA transcription can involve multiple RNA polymerase in a single DNA template and multiple rounds of transcription, which is called amplification. So many mRNA molecules can be rapidly produced from a single copy of gene. That means this one sequence is completed, then RNA polymerase will restart from here. So multiple copy of mRNA are produced so that the multiple RNAs are produced for the manufacturing of proteins. So many mRNA molecules can be rapidly produced from a single copy of gene, which is located in the entire sequence of gene. Elongation also involves a proofreading mechanism that can replace incorrectly incorporated bases. So this is also one of the proofreading is done during the elongation, and then termination. There is a code or the enzyme available which terminates the elongation of the messenger RNA in the making the copy. Bacteria use 2 different strategies for transcription termination, which we will not go in detail because that is not important from our point of view. RHO independent and RHO dependent. Now let us see genetic codes. mRNA is manufactured There is a question, how to identify gene of interest. So we have started with what? We know what protein we want to manufacture. We know what is the amino acid sequencing. And from amino acid sequencing, now we will come to know which mRNA is required from this genetic codes. Once we will know this genetic codes, we will be able to reconnect genetic sequencing and the amino acid sequencing. The code is universal. All prokaryotic, eukaryotic organisms use the same codon to specify each amino acid. So now the messenger RNA is made up of nucleotides, where we have 4 nucleotides, A, U, C, G. Now these 4 bases, they form a genetic code. And each code is responsible for 1 amino acid out of 20. Let us see what is this genetic code. The code is triplet. 3 nucleotides make 1 code. 61 of them code for amino acids. 3 like UAA, U and UGA are nonsense codes or chain termination codes. So these 3 nucleotides will make 1 genetic code, and each genetic code is responsible for 1 amino acid. The code is degenerated for a particular amino acid. More than 1 word can be used. The code is non-overlapping. A base in mRNA is not used for 2 different codons. The code is commaless. There is no special signal or commas between the codes. The code is non-ambiguous. A particular codon will always code for the same amino acid wherever it is found. Now let us see the codes. Now you can see this is our mRNA. So this is 1 nucleotide, this is 2nd nucleotide, and this is 1 of the 4, 1 nucleotide will be there. So if I see here U, this code will produce phenylalanine. This is universal. Anywhere 3 U nucleotides are in sequence of messenger RNA, they will translate into phenylalanine. Similarly, UUC will also make phenylalanine. UUG or UUA will translate into leucine. The same way, all 21 amino acid codes arranged in the messenger. We have seen 3 nonsense. So these are the 2 nonsense. They will stop the translation. And the 3rd one is UGA. These 3 are called the nonsense codons. And these codons, those who are present here, we can see here, because we know U, T, C, G, we don't know, but yes, by this way, they will be are arranged in the messenger RNA, and this 1, 2, 3, they will make a code, and this codon translate into 1 amino acid as per this table. Any doubt in making? Now we have reached to preparation of amino acids. So no is as and when the particular protein is required system. And we have seen the tRNA and RNA are the part of transcription from DNA fragment to mRNA fragment. So mRNA fragment, rRNA fragment and tRNA, they are transcribed as and when they are required. Correct? What is the process to produce DNA vaccine for RNA virus? RNA virus, what it will do? RNA virus will infect our body, and RNA will what will happen? RNA will start translation and produce antigens, which are harmful for our body. Very good question, and Mr. Basu has asked. If we take RNA virus, RNA will go inside our body. It will go inside our cell. Inside this cell, it will start directly the translation. And the result of translation will be assembling of amino acids into a peptide or a protein, which will react as an antigen in the body and will start deteriorating the health. Our body will react to that antigen and will start manufacturing the antibodies to neutralize the antigen, which is developed by RNA virus. And that is why here one thing I will add. We all are talking about the booster dose. What is booster dose? Whenever we are taking a vaccine, like we can take example of attenuated vaccine or RNA virus vaccine, they all are manufacturing the antigen, which is a protein. And the process is the same process adopted by the foreign RNA or DNA or a viral cell as per the central dogma, DNA to RNA to protein. Now, when we have been given a vaccine, our body has been forced to generate the antibodies of that antigen. And then central dogma process, DNA to RNA to protein for antibody, is recorded in our memory cells. To activate this, memory cells are T cells. These to activate or reinforce that memory in our body, that booster dose of vaccines are given. So I have diverted because I have got this idea during this. So the function of RNA virus I have explained. This will go inside the body. It will start translating immediately, produce antigen in the form of proteins. And that protein is a toxin for us. And our body's immune system will start responding to that toxin. Immediately, the information against that toxin stored in our DNA will be activated, opened up, prepare a messenger RNA. Messenger RNA will be transferred to ribosomal RNA, and ribosomal RNA will manufacture the protein, which is a antibody for that RNA virus. The same way, the between DNA and RNA vaccine, if the DNA in our body, it will go inside our cell, it will open up, it will make mRNA, tRNA, rRNA, and then start manufacturing the antibody. In DNA, the DNA vaccine, there is one step, that is the transcription is added. In RNA vaccine, transcription will not be there, only translation will be there to produce the antibodies. Okay, so the protein synthesis translation proceeds in 4 phases at ribosomes, because the ribosomes are the sites where the proteins are manufactured. Ribosomes are located on the surface of endoplasmic reticulum, which is called as the powerhouse, where a lot of ATPs are stored. So first is activation of mRNA, then initiation to start this coding, this one, and then elongation. What we have seen in mRNA, the same function, but there what we have assembled the nucleotides. Here what we will assemble in translation, based on these codes, we will be assembling these amino acids and jointing together to make a polypeptide or peptide or protein, which is a antibody. Activation, initiation, elongation, and termination of manufacturing of a protein, which is a monoclonal antibody, starting and ending. This is the protein synthesis. Any questions? Please feel free, because we have completed a lot of things. Prasanna, we will take a break of 15 minutes. And if any questions are there on protein sequencing, N-terminal, C-terminal, G sequencing, any confusion how the monoclonal antibodies are manufactured in the biopharmaceutical industry. Prasanna is there or not?
Yeah.
Prasanna, anybody is having question? Because on chat box I have already answered. If anybody is having question, we can have a quick question answer round on this amino acid sequencing and that gene sequencing and the
Right,
protein
sir.
synthesis.
Yes,
Otherwise, we can break for a tea break quickly.
Yeah, let's have a tea
15
break for 15 minutes, I guess. And then
yes.
we will start with the process development, right?
16:30 we will start.
Yes, yes.
Right.
Okay.
Right.
Okay.
Done, sir.
Prasanna, if everybody is here, we can start now.
Let me check, sir. I think it's 4:29 already. Yeah. Maybe within a minute we can start, sir. No issues.
Okay. So till now what we have seen that the amino acid sequencing, both N-terminal, C-terminal, then the DNA sequencing, then codons and the synthesis of desired protein. Now there are certain aspects how these things are done in the lab. These clones are manufactured. So once we have the structure, we know structure of our protein, amino acid sequence, and we have the corresponding DNA sequence also. We have reconstructed this DNA sequence, and then only this DNA will not be able to manufacture the protein. So what we have to do, because DNA is a very small fragment of entire DNA, which the part which is translating, responsible for manufacturing or translation of the desired protein. So something is required which can carry this small fragment inside the cell. Because in the lab or in the manufacturing, ultimately what we are going to do, live cells, whether it is a bacteria, fungi, or animal cell line, they are live cells, and we provide the suitable nutrients, salts, ingredients, temperature, oxygen or carbon dioxide, suitable environment so that these cells should grow. And when they grow, the process of this transcription and translation happens in the cell. And that is why the protein of interest is getting manufactured in large quantities. Sometimes what we do in a cell, which is recombinant cell, we call multiple copies of this DNA sequence we insert inside the cell, so that the multiple copies will manufacture multiple copies of protein, and our yield is increased. So for carrying this DNA fragment, we need a vector which can take this DNA along with this inside the cell. So these are called vectors. So vectors serve as carrier to allow replication of recombinant DNAs. So in this vector, there are three things we have to do. Origin of replication. Let me take the torch. 1 minute. Okay. So the origin of replication in the vector which is taking inside this DNA, there should be a origin of replication. Multiple cloning site, as I told you, multiple copies of this DNA are inserted into the vector, so that at a time when replication is happening, that multiple time that protein is manufactured and the selection of gene. So there are majorly three kind of vectors used: plasmids, PBR322 PUC vector, commonly used vector. Then phages are used as a vector, phage cosmid M13, and phasmides are used as a vector. So let us see how these vectors are looking like. This is a very small plasmid. The plasmid PBR322 showing the locations of 11 unique restriction sites that can be used to insert foreign gene. So now if you see, this is the vector for E. coli. The locations of the two antibiotic resistance genes, this is ampicillin and this is tetracycline. If we want to insert ampicillin gene, we can insert here. If we want to insert tetracycline gene, we can insert here. What are these genes? These genes are responsible for the immunity to these antibiotics. That means if this gene, ampicillin gene, is inserted here and this vector is going inside a cell, that cell will be resistant to ampicillin. Similarly, if we insert tetracycline resistant gene in this vector and this vector is inserted inside the cell, this cell will be resistant to tetracycline. That is why the world is worried about the resistance developing towards the antibiotics, because our genome is developing or modifying certain areas of genes which are making us resistant to ampicillin or tetracycline or other antibiotics. And these are in the kilo base pairs. Kilo base pairs, again, 1 kilo base pair is equals to 1000 hydrogen atoms weight. So this is called the vector. Now let us see, this is the origin, what we have seen. Now let us see how this is done. This is our vector, ampicillin gene and tetracycline gene, and this is our gene of interest, which we have reconstructed. What we do, with the help of enzymes, we cut both of them. We cut here also and we cut here also, so that the grooves corresponding to each other are made. Or you can call it that the amino acid here or the nucleotide here or the nucleotide here, they can join together here with the help of enzymes. When we join these two fragments, this is a vector and this is a gene of interest. Ligase means jodne wala. We use this enzyme and this fragment is joined together with this vector. Now this vector is prepared for us. Now this vector is inserted inside the cells. You see, this vector, this is E. coli cell, this vector is inserted inside this cell. And there are many procedures through which this insertion is happening in the lab. Now in the lab, you see this, this is the linear phase of the DNA. We have cut it and we have, this is our vector, and we have cut this vector also, and this yellow, blue, we have combined this completely, and this is the straight DNA which has gone inside this multiple copy. You can see 6 copies has been inserted inside the cell. Now when this cell is growing, when it is doubling, this vector is also doubling and producing the protein of interest. And then protein of interest inside these cells is raptured, which we have called lysis before centrifugation. The product is liberated in the medium. So this is the process happening in the lab to develop a cell which is having the vector along with gene of interest, which can do the transcription and translation to manufacture the protein. Now, how to check? When we do this insertion, it may be, or we are combining these 2 fragments, it may happen that certain cells are doing well, certain cells are not doing well, but they are inserted inside the cell. So for removal of non-performing cells, what we do, this is a original plate where you can see 5 colonies. We take a replica plate, this is a cushion, and we take the footprint of these 5 colonies on this replica plate, and then we again make a replica on the blank plate. So these all 5 plates, these 5 colonies, they have come into this replica plate, and then again they are transferred into a plate and 5s are going. Now in second case, when we have inserted a tetracycline resistant gene in the vector. So what will happen? The colonies, those who are performing well, they will not grow. So in this original tetracycline plate, you can see, for example, this is a tetracycline resistant colonies, and we have inserted the gene for ampicillin resistant. So you can see in second plate, these 3 colonies has not grown. That means in these 3 colonies, that ampicillin gene is not inserted properly. That is why when this ampicillin is present in this media, these 3 colonies, dotted one, are killed. And rest all the colonies, they are having ampicillin resistant gene inserted like this. And they are resistant to ampicillin and they are growing when the ampicillin is present in this media. So by this way, the colonies which are grown, they are called RCB, Research Cell Bank. We take these colonies in different tubes from one of the colonies which is performing best. How to test the best performing? We test all these performing colonies with increasing concentration of ampicillin. So those who are having the enough capacity to resistance ampicillin, they will grow to the highest concentration. And that most performing colony will be taken as a research cell bank. All will be research cell bank, but that most performing will be taken as a final research cell bank, will be used for the manufacturing of MCB, master cell bank. And that master cell bank, from that master cell bank, one of the vial will be taken to prepare WCB. So this is the simple procedure adopted in the lab to select the best performing clone for taking to the manufacturing. So this is what we have seen. Now come to when we are ready with our clone, that the fragment DNA which is responsible for our protein is inserted into vector and then transferred inside the cycle. Now we have to optimize the growth of the cell. We have seen the expression of the gene which we have inserted, that it is quite nicely expressing the function for which it has been inserted inside the cloned inside the cell. Now our objective is that the cells, when we are growing, they should grow to optimum condition so that the maximum product can be manufactured. So there are certain things. First thing is the media in which these cells are grown. So for growth of the cell or human body, whatever it is, living organism, we need carbon source for energy, nitrogen source for energy, some inorganic salts and trace elements. So different combination of carbon, nitrogen, inorganic salt and trace elements, they are tested with this selected clone. And wherever, in which composition our clone is performing the best, is selected for as a media for the manufacturing. So this is the second stage where we do the media optimization for maximizing the production, and then for optimal growth of recombinant cell and production of the protein of interest. So this is what involved in the development of process, starting from our interest in which protein. Correct? Now, if you have any questions, please feel free to have a question answer session, because next is the process development. We have developed our clone. We know our protein structure, amino acid sequence. We know the gene sequence. We have constructed the gene of interest. We have conjugated the gene along with the vector, and the vector is inserted inside the cell. And then we have selected the best performing cell. And after best performing cell, we have optimized the media for growth of the cell. From this is done in the lab, an R&D lab. And generally these experiments are done using the single-use flask and tubes, tips, molecular biology apparatus, shakers, and sometimes filtration equipments. After that, this will go to the process. Media is optimized. Now process conditions like what should be the pH, what should be the RPM, what should be the dissolved oxygen concentration, to maximize the reproduction of cell and production of protein of interest. So that is called as a process development. Yes, I have media optimization is done in lab. Yes, media optimization is done in lab. I am not sharing my screen right now, because this was the end of that molecular biology development part. So any questions on this part?
I think there are no questions, so we can go ahead with the next module, definitely.
Prasanna, what I am thinking, because I have an urgent call from Biocon, because one of their change the view option more. Okay. Not the question for me. They wanted me, because their one process development is going on, so they have scheduled a call from 5:30.
Okay.
And next topic will be more than 1.5 hours. So we can start tomorrow morning and we can extend tomorrow until the entire workshop is completed.
Let me check, because just hold on for a minute. So because tomorrow we'll be also having similar sessions, right? 9:30 to 5:30. That's the plan tomorrow. Okay.
If Dr. Saxena is having call, then there is no choice, right?
Actually, Piyush, what happened now? There is one process development for phase 3 is going on. So they have got some issue in that. So they wanted a call with me. 5:30 they wanted, because 5:30 US team will also join.
Okay.
No, Dr. Saxena, we are fine with that. So that's okay. So probably, Prasanna, before we start tomorrow morning, so you can catch up with Dr. Saxena and just fix on the overall priorities which we can cover, first in the morning, so that we are aligned.
Okay.
Okay.
And then we can cover the rest of the part.
Okay, because tomorrow we will save a lot of time of introduction and the evaluation part. So
right.
Tomorrow we will have more time, and if required, we can extend in the evening, because tomorrow I am not having any assignments.
Yeah, sure.
Sure, no problem.
Okay, so Prasanna, you can distribute today's slide decks to everybody, so that in the evening they can go through these slides, and if they have still having some questions in the morning hours, we can discuss all these questions and doubts, and we will proceed for the process development.
Right, sir. Okay.
Okay. Thanks.
Okay.
Sure. Thanks.
Okay. Thank you very much, Prasanna and Piyush. We'll meet tomorrow morning 9:30. And thank you, everybody.
Okay, thank you. Thank you, sir.
Thank you, sir.
Thank you very much.
Thank you, sir.
Thank you, sir.
Thank you.
Thank you, sir. Thanks, everyone.
Thank you, sir.
Thank you, sir.
तो बंद कर दो ना अभी, लीव एंड मीटिंग कर दो ना अभी।